हाँ जी सर, शुरू करिए जी, with your permission.
Yes, 2 minutes. I think 10. It's 10:48. Let's wait for 2 minutes.
Okay, sir.
Yes, please start.
Thank you, sir. Before we begin, I request all of you to please rise up for University Tuni. Requested respected Vice Chancellor Professor B.S. Kumanji, Dr. K. Thangaraj, Director, Center for DNA Fingerprinting and Diagnostic, Hyderabad, Dr. Sana from Loughborough University, UK, Dr. Vasda Sambyal from Guru Nanak Dev University, Amritsar, Dr. Rajinder Kaur, Head Department of Human Genetics, Dr. Ginjinder Kaur, and all the viewers and participants who have connected with us through this virtual platform. I heartily welcome all of you on this special occasion of Golden Jubilee webinar of the Department of Human Genetics. I'm feeling very happy to be a part of this department, which has grown now to 50 years. Long back, long back in May 1970, this department was started under the able guidance of Professor L.S. Sidhu as Department of Human Biology, which has grown, progressed, and now developed to Department of Human Genetics. I wish Dr. Sidhu could join us today, but because of his busy schedule, he has sent a recorded message of some of his experiences in this long journey. To commemorate this special occasion, we have organized a webinar today where three distinguished personalities, Dr. K. Thangaraj, Dr. Sarvjit Mastana, and Dr. Vasda Sambayal will enrich us with their expert opinions and thoughtful insights. Before I call upon head of the department, let us listen to the recorded message sent by Professor L.S. Sidhu, founder of this department.
Vice Chancellor, PUNJABI University PATIALA, Dr. V.S. Kumar, Head of the Department, Dr. Jubilee, Organizing Secretary, Dr. Paul Singh. It is a great pleasure for me to be with you on this day when we are celebrating the Golden Jubilee of the department. The department was actually started in 1970. The precise date is 11th May. And I was invited to start this department because at that time the university was starting other departments also. Botany was started in 1967. Zoology department was started in 1968. Then came the term of human biology. The Vice Chancellor at that time, Mr. Narang, thought it good to start a department of human biology. This was a new department. This was the only department at that time of human biology in the country. Because anthropology department was there in Delhi, and anthropology department was also there in our university. So he thought it that we should have a department which should study the biological aspect of people. Either we call it biological anthropology or human biology, so that you could study the biology of people of this region. Being Punjab being the border state, we want to study all aspects of the population of this state, not only Punjab state, Himachal, Jammu and Kashmir, and other adjoining areas. So it was in 1970 that I was invited, and I was allotted only one room of 2 bay to start this department. This one room was there where the human department is located now. Other rooms of that floor were occupied by departments like translation, linguistics, etc., and other official work. But gradually, we were given more space, and it was in the month of July or August that we started the first batch of students. We admitted the students, and I remember the number of students was 17 at that time. Even though 15 was the limit, but still more and more students were coming. They were thinking that this is a good department, this is a new department. We at that time did not have any infrastructure, we did not have any library, we did not have any practicals, we did not have any instruments to do practicals. So after starting the classes, I sometimes used to take the students to Punjab University, Chandigarh for the practicals, and we first of all thought of good syllabi so that the students should get good knowledge of anthropology. Because I had already come from Chandigarh, Punjab University, Chandigarh. I was lecturer there and joined here on 11th May. And before that, I had the honor, I had gone to Oxford University, Oxford, UK, for studies in human biology. So I was rather conversant with the total field of human biology. And looking at the local needs of needs of the country, of the region, at that time, we formulated the Bible. For first year, we had fundamentals of human biology, that was one paper, human anatomy, human physiology, and the other paper was human evolution, primates, and particularly with reference to Shivalics, because Shivalics were very near to there. Other paper was historic archaeology and social anthropology. This would give a fundamental of human biology because when you want to study human biology, you need to know all those things. So in second year, we have papers like human growth and development and body composition, human genetics, human ecology and adaptation, human variations. This was the syllabi, and there were practicals to be done in the first year and second year also. Field work was made necessary because without field work, these subjects like human biology and anthropology, they cannot go very far. So we made it compulsory for every student to go for a field work, to collect data, to analyze this data, and to submit in the form of presentation. I'm talking of those years, and perhaps the things are going on in the right direction now also. Now, if you look at the department as we started, I was the first to join to start the department, and gradually one more teacher joined us. His name is Dr. I.T.S. Vansan. And after some time, about a year or so, Dr. Kabir Singh also joined. And we were the three persons who carried on this department for a very long time. It is a coincidence to know that even now there are only three teachers in the department. There should have been at least 13 at the moment because the pace with which the department works, there is need to have more vacancies, more teachers for the department. And I must name two more persons who joined me during that time, near that time. Mr. Darshan Singh, he was artist, photographer and artist, and Mr. Devinder Singh, he was the clerk of the department. So with these five people, we started this department. And all of us worked very hard for a very long time to put this department on firm footing. Now, if I remember the names of the first batch of the students who had achieved good positions, first batch, Dr. D.P. Patnagar was there. He retired as professor and head of this department. Professor Mr. N.S. Mittal, he was also there with us for some time, but then subsequently he left for Canada, and now he is in USA. He is now heading a department there in USA. Dr. Praveen Singhal, Dr. Pushpa Singhal, they were other two persons. They also retired professor and head of the same department. Another person was Dr. D.K. Kansal of the same batch. He actually he was with us for quite some time, and then he left to join as principal of a Delhi University college. The second batch and third batch, there was Dr. S.P. Singh was there from the second batch, and Dr. Kamla Mehta from the third batch. And this happened, this went on gradually on and on. Other people also came and joined the department. They helped us in all the way. It will not be possible for me to name all of them, to give their names, but the students, I think, you know about them. Now, as the department was going on, it was in 1972 that we got a research center in sports. And this was in the years 1972 that UGC gave us a center in which the UGC gave us 4 positions to work on, to, to do research on sports, theology of sports, biochemistry of sports, morphology of sports, and yoga and sports. Yoga at that time, 50 years back, about 50 years back, we started doing yoga and sports, and now yoga has become very popular these days. And 4 people joined us. The name of this person named also Mr. S.K. Verma, joined geology, he came from Delhi. Mr. M.S. Sohol, he was biochemist, completed as a different as our student. Dr. J.L. Knott, also from Delhi, came to join us. He started working in physiology and yoga. This actually center came to us, and Dr. Mr. at that time, Mr. S.R. Mahindru, who retired as the principal of the Government College of Physical Education, he was appointed coordinator of the center. He knew about physical education, but for research in these fields, he did not know much. So we collaborated and we started this center. This center remained with us for quite some time, and the center did very good work. In 1990, this center became a full-fledged department of sports sciences. The Department of Sports Sciences already there doing very good work, and this is an offshoot from the Department of Biology. Subsequently, they started physiotherapy of sports person, physiotherapy in sports. And gradually the Department of Physiotherapy came out from the Department of Sports Sciences. So actually, Department of Human Biology has offshoots, and the Department of Therapy, Department of Sports Science, they are rather the children of the Department of Life. Now, looking at the PATIALA has the National Institute of Sports. So I established contact with the National Institute of Sports. There was Mr. R.N. Anand, Director of Sports at the time of the National Institute of Sports. He was very much interested in the scientific study of sports person. So one of our students, Dr. H.S. Sobhi, at that time Mr. H.S. Sobhi, he joined the department and we established a full-fledged department of, of sports medicine, as we call it, the faculty of sciences at the National Institute of Sports, and many of our students have gone there and they are doing very good work. So this is look at the scenario that human biology, the people from human biology, they started working on physical aspects of the population and started to work in the field of sports. Some of our students, one I would say, of the first batch, Dr. P.P. Singh, he started doing work in forensic studies, and he joined forensic study, and he was a very capable person. At the moment, he is a very big man in Delhi. He is the director of an institute in Delhi. So that is how the department started its work and gradually going on and on. And the department is by all leaps and bounds progressing. We, as we were 20 years old, an international conference was organized by the department in which delegates from most of the countries, I think 30 delegates from abroad, they participated, and it was a very good success of the department. As I look back, it seems that the Silver Jubilee year 1955, the department was at its peak. Then, at that time, most of the students from other universities would come to us for training, and we would give them training, and they would go back. This department was internationally, I would say, is internationally known, even now, doing good work. And we involved medical college also in our pursuit of doing work on biology. And gradually many students from medical college started coming to us for their MD, MS and other courses. So that is how we spread human biology to other fields also. I remember from nursing, many students from nursing came to us, they did their PhDs, and now they are principals or in higher positions in, in this area, job and other fields. Some of our students have gone to education, to biochemistry, to biotechnology, and many other fields. I may not at the moment may not be remembering all of them. So this is how the department progressed, and we are celebrating the Golden Jubilee. So at as we celebrate the Golden Jubilee, we must know what are our good points, what are our weaknesses. I have talked about some of the good points, but weaknesses we have also to talk about weaknesses. As the department progressed, we collaborated with the many international universities, international institutes. I will name some of them from United Kingdom, UK, because I had studied there at the University of Oxford, so we have very good collaboration with them. Department of Anthropology, University of Durham, Department of Human Genetics, University of Newcastle upon Tyne, Department of Biological Anthropology, Oxford University, Department of Human Biology, Dublin University. These are the departments of England. Then we have a very good relation with the Department of Human Biology, University of Bremen, West Germany. In the Bremen, West, what is his name? He would come here and give a lecture. I would go there, other people would go there and give a lecture. Then University, Department of Anthropology, University of Hamburg, West Germany. Institute of Human Genetics, Hamburg, West Germany. Department of Anthropology, Kiel University, Kiel, West Germany. Department of Anthropology, University of Ethiopian, Budapest, Hungary. We have very good collaboration in Hungary, where we collaborated for both studies and also for sports. And I said, Ivan, come here, no more. You will come here to us, and we will go there. And then Institute of Sports, Budapest, Department of Physiology, Simon Fraser University, Jubilee, British Columbia, Canada. University, Department of Physical Education, Winnipeg University, Winnipeg, Canada. Department of Physical Education, University of Edmonton, Canada. Department of Physical Education and Recreation, San Diego University, San Diego, US. There were other institutions in the USA, I can't name all of them, who have been coming to us. We have been going to them. So the department was at its peak. And most of us, we became very senior in the university, and myself and Dr. Sponsor, we were the Dean of Academic Affairs in the university, and I also acted as Vice Chancellor of the university for some time. So the department was very well known in the university itself, and also it was very well known all over India and international level. So this is Golden Jubilee, and 50 years is a very long time in somebody's, for somebody 50 years is a long time. For an institution, perhaps it is not that much time. But I think 50 years should be a good time, and institution grow and flourish and give good results and give this to the society. And it is a very rare occasion for me to be with you, to join this Golden Jubilee, because normally 50 years people don't survive 50 years. I have seen Department of Zoology, Department of Botany, they have not the heads of the founder heads of these institutions, these departments, they could not. And it is a very big God that blessed me to be with you for this time. It is my good wishes to the students. I was thinking that 48 batches of students must have passed about 50 years, 48 batches, and I was wondering, it is about nearing 1,000 students, most of them in most of the countries are there. Once I had a chance to go to New York, and in one institute I found 4 or 5 students doing very good work in New York. Wherever you go, you find the students, and I wish this department a great success, and I wish they will sit together now and find out if what else can be done, what how you can progress further for the education. You can progress in. Thank you very much.
I'm I'm very thankful to Professor Sidhu for sending such an interesting excerpts from his memories. I wish him long, healthy life. Now I request Dr. Jinder Kaur to please share her views and thoughts.
Thank you, Dr. Puneet. Uh, Very good morning, and Professor Vasudha Sambyal. Fingerprinting and Diagnostic, Hyderabad. Dr. Vasudha Sambyal, Professor Guru Nanak Dev University, Amritsar. Dr. Sarah and delight gold has been standard for of exemplary purity and excellence since times immemorial, and I cannot of this part to think about the future. The department was established in 1970 as a Department of Human Biology under the able leadership of Professor L.S. Sidhu with minimum infrastructure but maximum zeal and enthusiasm. Joined this department followed by Professor I.G.S. Bansal and Professor Lakbir Singh. The aim was to increase the prosperity of human biology and to search for new knowledge on the avenues of the subject. A UGC funded research center in physical education and sports was set up in 1973, which became an independent department of sports science in 1989. A human antigen research program was initiated in 1975 under the guidance of Professor P.K. Srivastava, and an animal house was built in a separate building. In this program, studies were conducted on different populations and to know the society. Half a century later, the objective as set up by the founding fathers has not changed. Remain as a beacon for others to follow. Each one of us has faithfully and satisfactorily worked toward the ideals set up by our founders. Later in 2000, leadership of Professor Satbir Kaur for the better placement of students and explanation. The advances made in our field have contributed symbolically to the well-being of the humankind. The contemporary dynamism raises the merit and utility of research and research in human genetics, the department has taken up various ventures in research over the years. This is an extensive ongoing research on the genetic investigations of molecular characterizations of human cancer, human growth and development, nutrigenomics of complex diseases, and autoimmune disorders. Major and minor research projects are DST. The department has a rich history of providing supervision to MD, MSc, and a comprehensive doctor research program in the field of immunogenetics, human population genetics, biochemical genetics, cytogenetics. Molecule was identified with this financially support, the department has further strengthened teaching and research by setting up a centralized research laboratory which house major equipment including fluorescent microscope for assessment and documentation system, biochemistry analyzer, ELISA students placed in research centers, pharmaceutical companies, in health system, and in educational institutes like schools, colleges, and course that lies ahead. It is therefore our responsibility to scrutinize our journey and up an initiative to commence postgraduate diploma in genetic counseling this year in the same direction. We are also planning to collaborate with other institutes to widen the for the upgradation of the department. I once again congratulate all of you for this glorious journey of 50 years and also wish for many more milestones. Thank you, Dr. Puneet.
Thank you, Dr. Jinder. Now I request our Vice Chancellor Sahib to please grace this occasion with his presidential remarks.
Good morning, friends. Esteemed Professor L.S. Sidhuji, Dr. K. Thangaraj, Director CDF D. Hyderabad, Dr. Sarvjit Mastana, I know he joined from United Kingdom. Early in the morning he joined. Welcome and thank you very much, Dr. Sarvjit Mastana ji. We have esteemed ma'am, Dr. Vasudha Sambyal ji, Professor Rajinder Kaur, head of the department, Dr. Puneet Palji, convener of the celebrations, and dear colleagues, non-teaching staff, students, and alumni of the department. First of all, I extend you all very warm welcome and also take this opportunity to extend heartiest congratulations to the Department of Human Genetics on this historic day. Golden Jubilee Day is a historic day in the life profile of a department, institution, as well as individual, particularly married couples. Therefore, this day was eagerly awaited by the Department of Human Genetics. Today, this day has happened, and I once again extend this department heartiest congratulations and wish this department to scale greater heights in future. Friends, when we were listening to Dr. Professor L.S. Sidhu, it was a delightful experience, very rewarding experience. It was an intellectual feast which has no parallel till now. I have never seen, when I was listening to him, frankly speaking, believe me, I could not believe that in 1970, the young professor who was addressing us now, Dr. L.S. Sidhu, was the founder head of the department. His age, his energy, his hard work, all together suggest he is as young as he was in 1970 when he established this department. Therefore, my heartiest compliments to him. My heartiest compliments to him for this energy, enthusiasm, zeal, and hard work. See, friends, when we were listening to him, see, these are the historic days. I mean, I will come to Professor Rajinder Kaur as well later on. But when we start with the founder and see the hardships he and his team, Professor Ais Bansal and Professor Lakhbir Singh, they faced. See, now we are reaping the dividends of their hard work. We cannot imagine that they started with one room. Now, at present, each faculty member needs a separate room, but the department was started with a single room. Imagine this situation. Imagine the situation. When I was listening to, I've listened to him very, very carefully. I've left the structured lecture which was prepared. I'm not going to read any sentence from this structured lecture, but he motivated me to speak on these lines. Starting with one room, no infrastructure at all. See, can I convince my any colleague now? I can't succeed in that mission now. That, dear friend, even if you are not having infrastructure, even if you are having a single room deliver, they will say, sorry, sir. See, therefore, let us get inspiration from Dr. L.S. Sidhu and his team. I know I have met Professor Bansal a couple of times. I am not able to recollect whether I have met Lakhbir Singh or not. See the energy. Still, they are young in their delivery. Therefore, when we were enjoying the historic tour, Professor Rajinder, Professor Dr. Puneet Pal Singh, have you seen the notes he has prepared? Have you seen the charts he has prepared? Have you seen each aspect he has dealt with thoroughly? If we get anything from him and his team to become thorough in our profession, I think this department has no limit, sky is the limit. Therefore, you have a strong founder. You have a very strong team of founders. I think we are reaping the dividends of that foundation, but simultaneously, Professor Rajinder or Dr. Puneet Pal Singh and other colleagues who have joined, we have to take this department forward. You have very high profile achievements to your credit. When I was listening to Professor Sidhu as well as Professor Rajinder Kaur, this department is second to none in the university. But at the same time, to maintain that level and to improve further, when he was saying that I went to New York and I met my students. See, your alumni are spread throughout the world. Your ambassadors are throughout the world, though formally not appointed. Now, if you have that much ambassadorship throughout the world, then let us connect ourselves with them. Let us connect ourselves 70, to today, 50 years journey. Therefore, we have a huge resource knowledge base and what we call it social capital, networking, that when we connect ourselves with the alumni, social capital, Professor Rajinder Kaur, you will generate. That social capital would be source of development of your department. You will get huge support, unexpectedly huge support. Need is to approach them. Needs is to involve them. Need is to motivate them to visit the department, to come to the department and involve them in the development of the department. Their partnership, I think, hold the key so far future of the department is concerned. When I was listening to Professor Sidhu as well as Professor Rajinder Kaur, Dr. Rajinder Kaur, then they shared with us the way department, starting from a humble beginning of one room, 17 students, now is one of the leading department, having received huge grant, both major and minor project of all the funding agencies within India. And when I was looking at the program they are offering, the type of research, interdisciplinary research the department is undertaking, that's the future of this department. That's the 21st century, belongs to interdisciplinary research. Continue with the spree of interdisciplinary research. When I was listening to Professor Sidhu, that this department, human biology, now known as human genetics, is feeding to a lot many disciplines. Therefore, you are a discipline which is feeding to other discipline. Therefore, your responsibility becomes more than the single disciplinary sort of a department. Therefore, you have to deliver, Professor Dr. Rajinder Kaur, more than other department, because the foundation you laid down, the starting is made from you. When Professor Rajinder, Dr. Rajinder and colleagues, I was looking at your collaborations, and which Dr. Sidhu also shared. When Dr. Sidhu was sharing, I think he didn't leave any university of United Kingdom which he has not referred. When he referred to Newcastle, we had a project with Newcastle University. Then immediately I get connected with that university. Therefore, when he was listing, see, your history is very rich, ma'am. History we teach as a discipline so that we can use it in present and use it for future. Therefore, today, one lesson, if I have learned from Professor Sidhu's historic tour, is that our history, our heritage is so rich. Let us build upon that heritage. Let's survive all those linkages which this department had. You may not succeed 100%. You may succeed 50%. Today is, say, 25th. Today is our 5th Golden Jubilee. During this, Madam Rajinder Kaur, I can give you one responsibility, that during this Golden Jubilee celebration year, get connected with all institutions across the globe within India with whom you had collaborations. Okay? That would give you a sense of achievement. Those universities and department will also realize that here is Dr. Rajinder Kaur and her team reconnecting us with the past, connecting us with the past, and you can prepare a very good document that when you were having a collaboration with Newcastle upon Tyne, Newcastle University, what did you achieve? What did you achieve with other university? Even preparing that document, collaborator research, past and present, I think you can produce a wonderful document by revisiting and then connecting. Then that would be a source of inspiration for you, your colleagues, and future students. When I was having a look upon your collaboration, I think you have a very, very rich collaborative approach with the reputed national and international research organizations. Need is to revive those. Need is to revive or work with, because this 21st century is a century of collaborations. It's a century of knowledge clusters. Until and unless, see, no single department can deliver, no single individual can deliver, no single organization can deliver. You have to pool resources. COVID-19 has given us a lot many lessons. That if we have a situation like COVID-19, joining hand with each other, social sciences, health organizations, you have to join public policy, you have to join hands with each other, only then you can mitigate COVID-19. Therefore, COVID-19 has taught us a lot many lessons in the institution of higher learning. Therefore, let me conclude by saying that after listening to Professor Sidhu, I'm hugely motivated to meet him as early as possible and sit along with him and listen to him, the story, And if, ma'am, you can go to him, record the proceedings, and then get those typed and get those published, that would be a very good. Not only him, the other two colleagues, Lakhvir Singh and Professor Bansal as well, and so on and so forth. I think you go to them, listen to them, record the proceedings, publish those proceedings. I think excellent job you will do for the department as well as for the university. I know you are eagerly waiting for listening to Dr. K. Thangraj ji and Dr. Sarvjit Masthana ji, waiting from since morning, and Dr. Vasudha Sambhal ji. You are very eagerly waiting for them. I once again, ma'am, Dr. Rajinder Kaur, your colleagues, Dr. Puneet Pal Singh and others, I once again extend you all heartiest congratulations on your Golden Jubilee celebration starting from today, and express my deep gratitude to Dr. L.S. Sidhu, who started the proceeding with a note that the concluding day should be higher than the inaugural day. Therefore, Dr. Rajinder Kaur, tough job is ahead. That today the benchmarking has become so high that you have to build brick by brick. On the concluding day, you have to deliver in such a manner that we should also enjoy the deliberation on the concluding day. Thank you very much for giving me an opportunity to share my unstructured thoughts with the audience, and once again wish you heartiest congratulations, and also I wish the department to scale greater heights in future. Thank you very much.
Thank you very much, sir, for your motivational thoughts, blessings, and words of appreciation. We assure you that we will work hard and will rise up to your expectations. Now, moving forward, today we have a very eminent scientist with us, Dr. K. Thangaraj, and I am feeling very proud and privileged to introduce Dr. Thangaraj to all of you. Dr. K. Thangaraj is the Director of Center of DNA Fingerprinting and Diagnostics at Hyderabad. Prior to this, he was Chief Scientist and Group Leader at the Center for Cellular and Molecular Biology, Hyderabad. His main research interests are origin of modern human, cardiovascular disease, mitochondrial disorders, and male infertility. He has published over 250 research articles. He is an elected fellow of Indian Academy of Sciences, Indian National Science Academy, and National Academy of Sciences. He is recipient of J.C. Bose Fellowship, Sun Pharma Research Award in Medical Sciences, Raman Research Fellowship, Lifetime Achievement Award Excellence in Science, Distinguished Scientist Award, Dr. Subhash Mukherjee Oration Award, Professor Manjeet Singh Oration Award, Dr. Palle Ramarao Award, Professor G.K. Manna Memorial Lecture Award, Professor G. Jayaraman Oration Award, etc. He is a board editor of Mitochondrion, associate editor of BMC Medical Genetics, BMC Genetics, Tropical Medicine and International Health, and member of the editorial board of the journals Scientific Reports, Human Genetics, and Clinical Genetics. He was the president of the Indian Society of Human Genetics from 2011 to 2015 and the founder of the Society for Mitochondrial Research and Medicine. Now I request Dr. Thangaraj to please deliver his talk.
So good morning to all of you. Thank you, Professor Puneet Singh, for the invitation as well as introducing me. Honorable Vice Chancellor of Punjab University, Professor Rajinder Kaur, Professor Masthana, Professor Vasudha, and other colleagues from Department of Human Genetics, and students. It's indeed a great pleasure for me to be associated with this very memorable event of Golden Jubilee celebration of the department. It's not easy, as I've listened both Professor Sidhu and the Vice Chancellor, that how things moves, particularly 50 years back. It's very mind-boggling to see that how this department has evolved and contributed to the Human Genetics Society. In fact, I am indeed very much happy that as a human genetics students, I was from Department of Human Genetics, Madras University. In fact, I just came to know that these two departments have started at the same time. Department of Human Genetics at Madras University was started in the year 1969, and this is 1970. So it's indeed a double pleasure for me to be associated with this event and deliver a talk. So with that, I'll go on to share my slide. Is it visible now? Is my slide is okay visible?
Yes, it is visible, sir.
Okay, so I'll
सर
go
दिख
ahead.
रही है, ठीक है सर. It's
Okay,
visible.
Yeah, thank you so much. So as a human geneticist and population geneticist, I would like to take again the story which I've been associated with the last 2.5 decades. As one of my very interesting field of human genetics to understand the origin of modern human migration and connecting that with our health and diseases. To give you some idea that how one can connect with the evolution and disease, I wanted to show the slide where Professor Dibzhansky is one of the well-known evolutionary biologists. He wrote an article in a journal called the American Biology Teacher. The title of his article was "Nothing in Biology Makes Sense Except in the Light of Evolution." So how strong this message is. So this gives the idea that how important to understand the evolution to understand the human diseases. That's what I will get to know towards the end of my talk. So we all know that the human and our close relative in terms of evolution, chimpanzee, have diverged from a common ancestor called hominids about 6 to 7 million years ago. And what happened during the last 6 to 7 million years? There are a lot of anatomical changes. There are a lot of ability and so a lot of other aspects. To look into that carefully, one of the aspect is the ability to walk with the two legs, called as bipedalism. And the other one is ability to make tools for survival. And the third one, very interesting, is that the brain size of the human during the course of evolution evolved and increased in size. If you look at the early species, chimpanzees, the brain size was as similar as chimpanzee. Then slowly it has increased to 35% of modern human in Australopithecus. In Homo habilis, 50%. In Homo ergaster, 60%, and gradually increased. And it was predicted that in Homo neanderthals, it was predicted that the brain size was slightly larger than the modern human. But this species is no more. But we don't know whether the Neanderthal was very intelligent than human. But why did the species vanish? There are many hypotheses. One of the interesting hypotheses given by Paul Mullers from Cambridge. He wrote in one of his articles mentioning that the inherent violent behavior of modern human probably responsible for vanishing of the species. Nobody knows whether it's true or not, but possible, right? Then just to give you some example about the studies which was carried out on Neanderthals. One of the study where I'm also part of it, a few years ago we published this by sequencing 300 human genome all over the world, where you see the dots. These are the places the samples have been collected, and we have shown that all out of Africa humans carried up to 3% of Neanderthal genome. So in Africans, you don't see the black one, whereas different frequency up to 3% was found in other part of the world. But now recently, Seventh Fabo and his group have connected the Neanderthal genome, which was there in people who have migrated out of Africa, and connecting that with COVID infection. Somebody want study the full paper, they can take the reference from here and study. So I don't want to spend more time because we have a lot more slides to talk about. Then coming to the origin of modern human, this is well-known fact that the modern human originated from Africa about 160 to 200,000 years back. And very recently, in October 2019, there was another paper by looking at the ancient DNA, ancient samples, and they have pinpointed that the modern human must have arose in southern part of Africa. This is just confirming the existing theory of origin of modern human. Then why did they go out of Africa? And there are many specialists in different fields, including archaeologists, are working on this field to understand why did the modern human migrated out of Africa. So this paper, which published in PNAS, where the geologists have identified that the water level in a lake called Malawi was gone down up to 95% during the year between 135 to 75,000 years. Therefore, there's no water, there's no food for them to have. Therefore, they have started migrating out of Africa. And the question is, which route did they take from Africa, from southern part of Africa? There were again two hypotheses. That one of them suggests that the modern human might have taken the northern land route, because this is a land connection, people must have migrated towards north. But other group don't believe on that, because the climatic condition in Europe might not have favoring this migration. Even today, it's very cold, but you can imagine several thousands of years back whether it was possible to migrate in that direction. So therefore, the alternative hypothesis was that the modern human might have taken the southern coastal route. These are all based on the archaeological evidence present in these places. There's evidence in Australia suggesting that the modern human must have arrived in Australia about 50,000 years back. In China, 39,000 years back, Middle East, 47,000 years back, and in Europe, about 40,000 years back. But unfortunately, we don't have any very good archaeological evidence to suggest that when did the modern human arrived in Indian subcontinent. So although we don't have very good archaeological evidence, but there are some evidence to suggest that probably the modern human must have lived in this particular place. There's a place called Jolapuram in Andhra Pradesh. There was no biological material. However, they found the tools which probably the modern human might have used. So that's a place where there was excavation, and these are the stone tools have been identified in that particular region. And very recently, in a southernmost part of India, a place called Kiradi near Madurai. So they have been excavating, the state archaeological department, but recently they found two complete skeleton, and we are trying to analyze this genome and come to some kind of conclusion. But unfortunately, this bone is not very old. It could be maybe some 2 to 3 century old only. Similarly, another place called Adichanallur, again, this is in southern part of India near Madurai. The Central Archaeological Survey of India have excavated and collected some bones which we have recently obtained from them, and the work is going on. I don't know whether we could be able to address anything from there because the bone was not properly preserved. But where we were very successful was from Rakhigarhi, where probably you're close to your place, the Deccan College and many others, the archaeologists have excavated this place and obtained a complete skeleton of a woman, and that helped us in collaboration with many people, and we have come to the conclusion. Again, this is not very old sample. It's about 3 to 4,000 BCE. So this is the one, and where we found this as a two genetic component. One is the ancestral, very ancestral Indian sample, Andaman tribes. I will come a little bit later about that. And the remaining majority of them have come from Iranian hunter-gatherers, not from Iranian farmers or herders. So this is very old, but when talk about its age, it's only 3,000 to 4,000 BC. So again, it's not going to give anything about origin of modern human or migration of modern human in Indian subcontinent. But what it gave, another interesting point is that probably because we don't have Iranian farmers genetic component here, the agriculture must have started independently in Indian subcontinent. So all these study is not actually contributing to the question that when did the modern human arrived in Indian subcontinent. There was another study. Majority of the samples have come from Central Asia and South Asia, but except in India, where we have collaborated with a large number of people around the world, and the main person is David Reach. There are 500 and some bone sample. That's one of the largest ancient DNA study. So where we, again, it's not really giving any solid conclusion about the origin of the first migrant modern human. But so what it is giving is that there was some connection about the migration, the second wave of migration from Africa. But the people who have come from Central Asia, particularly the Yamana migrants, so they have come to northern part of India, but not early. It's very recently in terms of the age. It's 2000 to 1000 BCE only, but not very old. Same way, they also migrated towards Europe. Probably they are the people who have spread the Indo-European language, and but it's not giving any clue about origin of human in Indian subcontinent. So, however, although we don't have very strong archaeological evidence, but interestingly, that we have the population who maintained the endogamy for a very long period, we thought analyzing their genome would actually give information about origin of modern human in Indian subcontinent or migration of modern human in Indian subcontinent. So having that much of anthropologically well-defined population groups, it's not exaggeration to say that India is an ethnic museum, because we have collection of large number of human ethnic groups. So what is another interesting aspect is that these populations are well stratified in many ways. So one of the broad stratification is based on the language. In India, as most of you are aware, there are 4 major linguistic groups. Dravidians is one of the early language, and there are 5 states which speaks Dravidian languages, 4 languages: Tamil, Malayalam, Kannada, and Telugu. Austroasiatic also considered to be one of the oldest, but not as old as Dravidians. Tibeto-Burmans, who are in northeast and northern part of India, speaks Tibeto-Burman language, which is also similar to Southeast Asian language. And majority of the Indians speak the Indo-European languages. So in addition to that, there are very isolated languages like Andamanis, Nihalis, and a few more. Okay, so this is based on the language. Again, under each language, we have several social stratifications. We have like a caste. With the caste, we have upper caste, middle caste, lower caste, tribes, primitive tribes, and so on. So all of them put together made India one of the very complex country. And I don't know how many of you are aware that in India we have more than 4,600 anthropologically well-defined population groups, which includes tribes, primitive tribes, and 36 of them still follow hunters and gatherers lifestyles, like the Andaman tribes, which I come in the next slides. So this makes so much complex, and in addition to that, there are several populations who migrated very recently, like Siddhis, Jewish, and many more of them, which again makes India as a very complex country. Having this background, so what I'm going to talk to you now is about origin and affinities of different ethnic groups in India and how the following of endogamy marrying within the group is going to have impact on our health and diseases. Let's first look at the origin and affinities. As I mentioned in the beginning, I've been working on this field for last 2.5 decades, and to our surprise, we came to know the first out of Africa human is in Indian subcontinent, or none other than the tribal populations of Andaman and Nicobar Islands. I don't know how many of you are aware, there's a population who typically look like Africans are in Indian subcontinent. They are particularly in Andaman and Nicobar Islands. To give you some idea about this, so there are 572 small islands, which is about 2,000 kilometers from Chennai as well as Calcutta. There are 6 tribal groups, indigenous groups. 4 of them typically look like Africans: Jarvas, Great Andamanis, Sentinelese, and Onge. Other 2 groups typically look like Southeast Asians, Nicobaris, and Champagne. Among these 4 groups, Sentinelese are still in isolation. Nobody can go near them. They will shoot with arrow. Whereas these 3 groups have some contact with the people who are in other islands. You can see their number is very, very small. Great Andaman is even less than 50, Onge 98, Java 200, and Sentinelese 250. Only Nicobaris are big in number, 20,000, whereas Champagnes are approximately again 180. There are several reasons why the population size is very less, but I believe one of the reasons could be when the Britishers captured India. So this is how they collect the samples, and that story, in fact, I had and I removed it because it requires a lot of time. When Britishers captured India, they wanted to construct the cellular jail to put all our freedom fighters. So that time they killed large number of these tribal people. After killing, I don't know what they thought, how important their skull and air samples. They collected their skull, and it has been kept in Cambridge Duckworth Museum. And in fact, that allowed us to collect some sample, analyze, and compare with the existing the tribal groups. So we collected sample from all these individuals, and there are several ways we looked at their DNA. we also looked at the Y chromosome, but now I am not talking about that. I am going to talk about the mitochondrial DNA, and to the students, why we used mitochondrial DNA. Mitochondrial DNA are inherited maternally. It generally doesn't undergo any recombination. It goes generation after generation maternally without any change. Therefore, we effectively use that to trace maternal lineage. And this also been used for ancient DNA study very effectively, because the copy number is very high and it's a small genome, circular, therefore doesn't undergo very fast degradation. So these are the reason we use the mitochondrial DNA initially, but of course now the whole genome sequence is the gold standard for any study. When we sequence this, the entire 16.5 kb mitochondrial DNA, and based on the mutation exists in the mitochondrial DNA, one can assign specific haplogroup to any sequence you have, because each haplogroup is defined by evolutionarily conserved mutations. As you see here in this parsimonious tree, these are L-derived haplogroups which are exclusively present in Africa, but you can also find that the people who migrated out of Africa in the recent past, and if they are unmixed with any other population, you can see that. That we have also seen in a population called Siddhis, who have migrated from Africa about 400 years back. As we acquire additional mutations, those mutations should be evolutionarily conserved, not like the regular polymorphism. And those are the one we use it in assigning the haplogroup. That means every step of setup mutation will put them in different haplogroup, and you can see that different continent or different countries have different haplogroup. When we sequence the complete mitochondrial DNA of the Andamanis, we thought that probably based on their phenotype, they will fall into L-derived haplogroups, which is exclusively found in Africa, or early M haplogroup like M1. But to our surprise, they have additional mutation they acquired because they have been there for a very long time without mixing with any groups. Therefore, these mutations are very, very unique to the Andaman tribes. So these are the the position of the mutations. And based on the mutation, one can also calculate the age of the population. So based on this, we calculated that the age of the population is about 65,000 years back. So we concluded that the Andaman tribes are the first modern human who migrated out of Africa, taking southern coastal route and reached Andaman and Nicobar Islands about 65,000 to 70,000 years back. So this is one of the first, very first study using genetic tool to identify or to understand the early human migration. Of course, you can now connect that there are populations in southern part of India like Kurumba, and they share some of the genetic affinity with the Australian Aboriginals, which in fact connecting the early migration, which I mentioned earlier that the Australians have reached Australia about 50,000 years back, here 65,000 years back. Now we can actually have the connections. After this, of course, this is about 15,000 years, 15 years back, we published. Subsequently, after human genome was sequenced, there's a possibility of looking at the 1 million genetic variations across the human genome. Affymetrix and Illumina, they have a chip. So therefore, we used the nuclear genome, particularly the Affymetric chip, which has a 1 million SNPs. So here we carefully identify the population which represent all the linguistic groups, Dravidians, Indo-European, Austroasiatic, Tibeto-Burman, and the Manis, and also social group under each linguistic groups, so-called upper group, so-called middle, scheduled caste, and tribes, to have a representation from a large number of population in India. And we also used HapMap data set and human genome diversity panel. And there are several softwares which we use, particularly if you look at the PCA, which I'll show in the coming slide, where we have shown very clearly that Indians, where you see that there are large number of Indian groups, and these are the European HapMap samples, African HapMap sample, and Chinese HapMap sample, where you can see there are large number of individual clustering together, suggesting that these three groups, although samples have been collected from different part of Europe and Yorubans from different place of Africa, similarly Chinese, but they are genetically more homogeneous. That's why they are clustering together. That tells that India, where we have every single population, is unique because of the practice of endogamy for very long time, and whatever the mutation takes place in a particular group, that's going to remain in that group. Therefore, that makes every group as unique. Whereas here you see that there's no such difference in these, at least these three HapMap samples. Another interesting point is, although every population is unique, but there is some kind of genetic affinities between the groups. Otherwise, the cluster might have been somewhere here and there, but they are not forming a cluster outside this gradient. That tells that every population is unique, but still we have some kind of genetic affinities. So based on that, what we proposed was that the Andaman tribes who have migrated from Africa, that's a very deep-rooted lineage, while Andamanis were migrating through southern coastal route, we predicted that some groups must have stayed in southern part of India, whom we call as ancestral South India. So that's one founding population. Another one, there was a second wave of migration towards north, and there was a divergent somewhere in the middle, which you can connect with the paper which I mentioned about 500 ancient sample. And one group has gone to Europe, other group has come to northern part of India, whom we call as ancestral North India. This is old, but this may be much later. And these are the two founding groups in Indian subcontinent. After some time, these two groups have admixed and then gave rise to many population groups. So at that time, we are not aware that when this admixture would have taken place. To understand that, we took additional samples from south and from north. We wanted to have a good number of representation from ancestral South Indians and ancestral North Indians. So that tells that initially both ancestral South Indian and ancestral North Indians gave rise to many population groups, but these two groups have admixed between the last 2,000 to 4,000 years. What you see here, this is a kind of admixture which shown pictorially that there's a combination of the genome in different individuals. And if you recollect the PCA, where we have seen that there is a uniqueness, but there's also kind of affinity between the groups. But what happened for the last 2,000 years? Once the admixture took place, that stopped 2,000 years back, precisely 1,900 years back. Then every single population started maintaining their endogamy. So that tells that after the admixture, every population is now marrying within the group, which makes everybody, every population as unique. Okay, so this probably must have happened after the caste system arose and the social structure was formed. Therefore, every population very strictly following the endogamy. So now what I'm going to talk to you about is that how the endogamy is influencing our on our health and diseases. So in this case, now we don't want to see only Indian population. We wanted to extend this beyond India, particularly all the South Asian countries. And if you include that, now we have nearly 5,000 population. This is one of the very largest human diversity. Nowhere in the world you will find such a large human diversity. The reason why we took other South Asian population is that we have some background information to say that, so whatever we study in India, it's also true for other South Asian countries. So one of the reason is in this paper where we have shown that in the structure analysis, again using 1 million SNPs, in South Asia, you can see that there are two major genetic components. One is restricted to only within South Asia, but another one goes beyond South Asia, including Caucasus, Europe, and Middle East. But when we look at the compound, the genetic component, which is present only in South Asia, and it tells that actually India contributed about 50% of the genome to nearby South Asian countries. So this is one aspect. The second reason why we wanted to look at the South Asian was that when we identified a mutation 25 base per deletion in a gene called myosin binding protein C3 gene, this is in the intronic region. Students should know that we should not ignore the intronic mutations. What happened, this intronic mutation, it actually skips away one of the exon. Therefore, there's a truncation of the protein, which leads to sudden cardiac death, which leads, in fact, there are three different kind of cardiomyopathy. Cardiomyopathy is a heart muscle disease. One is dilated cardiomyopathy, where you can see the ventricle is enlarged beyond the normal size. Other one is hypertrophic cardiomyopathy, where the heart muscle, including interventricular septum, becoming very thick. The third one is restrictive cardiomyopathy, where the heart muscle becomes very stiff. Therefore, the heart muscle is unable to pump the blood properly. Hence, it leads to sudden cardiac death. If this mutation exists in the heterozygous condition, that means one copy of the chromosome has a mutation, other one is normal, then the individual survive up to 40 or 45 years without any symptom. But if it exists in the homozygous condition, and the individual die at very, very early age, less than 10 years, which we found by looking at the postmortem tissue of the children who died less than 10 years. In fact, we found 10% of children who died due to cardiac arrest are carrying this mutation in a homozygous condition. So the reason why I am saying is that when we try to look for the frequency of this mutation around the world and found that initially we looked at India, we found within India about 4% of the Indians carry this mutation in a heterozygous condition. When we try to look for the worldwide samples from 26 different countries, to our surprise, we found that the mutation is restricted to only South Asian countries, but not in other countries. Although the others have different mutation from the same gene, but this mutation, we predicted it arose in India about 33,000 years back. Now you can say that how it got spread, and you can recollect the work which I mentioned earlier, that there was admixture between different groups, people migrated. This happened much earlier than the endogamy practice happened. That's only 2,000 years back, but this is 33,000 years back. Therefore, it got well spread across India. This is just one example, and this shows that so whatever we study in India, either in terms of migration of people or in terms of genetic disease, that's going to be common among all the people in the South Asian countries. Therefore, we included people from other South Asian countries. So these are the places where we collected the samples, 2,800 individuals from 275 endogamous groups, and it tells that we have Dravidians, Indo-European, Austroasiatic, Tibeto-Burman, and also the Andamanis, Nigalis, and few other very isolated linguistic groups. We did several analyses, and one of the analysis which is very much relevant to our health is that when we try to look at the IBD score, identity by descent, and what it tells us is that about 1/3 of the population which we analyze are having high IBD score, identity by descent. So in simple terms, if you wanted to tell that if you take unrelated individual, 5 or 10 unrelated individual from the same ethnic groups, if they are really unrelated, then you will find, for example, if you are taking 10 kb region, then you may not find, you may find a lot of variation between the 10 individuals. As you expect that every 500 base pair, we may have SNP. Right? So if you don't see any difference between the 10 individuals from the same community, then you expect that they all have a same haplotype. That means they must have come from the same founder. Right? So that's what we identified, that majority of the population, although they say that they are unrelated, but genetically it looks like they are all from the same common founder. So that condition is actually high among most of the Indians, where I have kept the two references. One is Ashkenazi Jewish, and another one is Finnish population, the red color one, where you can see with the tiny IBD score, they have huge amount of genetic diseases. I'll show you in the coming slide. But imagine that most of the Indians are having several-fold higher IBD score, and you can imagine that what would be the consequences of those the high IBD score. And we predicted that a large number of Indians will have population-specific recessive disease, because once there's a mutation introduced, particularly we are not talking about the dominant mutation, we're talking about the recessive mutation. The dominant mutation, even if it's there in the one of the chromosome, that heterozygous condition is going to cause the disease. Here there's a hidden one. If it is the heterozygous condition, recessive mutation is not going to cause any disease, but is going to spread because of the marriage pattern in India. We marry within the group, therefore it's going to spread within that particular group. But after some time, it's going to be very high frequency, and there's a possibility of 2 individuals with the heterozygous mutations, they're going to marry, because they're going to marry within the group. Once that happens, naturally there will be children with homozygous mutation, therefore there's a disease. So that actually I put up in a cartoon on the cover, where you see that one half with mutation, that tells that it's a heterozygous mutation, and there's a possibility that individual with the 2 heterozygous mutation, they marry, the children with the homozygous mutation, therefore they all have the disease. On the other hand, the heterozygous mutation exists in many countries, but because the marriage pattern is not like Indians, they are going to marry within the group, and naturally he or she, when she marries, he or she is not going to find another individual going to have the similar mutation. Therefore, one can think that as a human geneticist, the students, you all have a lot more thing to do, because most of the Indians are, as per our study, going to carry mutation in heterozygous condition, which are of recessive in nature. Therefore, it's going to be a huge problem or burden for many populations. To give you that, how one can tell that, in this pedigree, if you look at, you consider that this is the mutation which is existing in the heterozygous condition. And I don't have to explain the genetic student, and wife and husband, they married, and they have 2 daughters. So both carry the mutation heterozygous condition. Again, both of them marry somebody from outside, therefore no problem in the next generation also, because the mutation is going in heterozygous condition generation after the generation. Now you see that after, this is just 4 or 5 generation, after 5 generation it goes like that within the community. Then there's a chance that these two individuals, after several generations, maybe they may not have their history of how many generations back they are related, but now they may say that they are unrelated, but they are from the same community, right? But now there's a possibility that these two individuals, when they marry, you can expect statistically 25% of children will have the mutation in homozygous condition. Therefore, there's a disease. So that is going. Now you can see I showed that there's Jewish and Finnish populations, and with the tiny IBD score, you can see that they have large number of disease, and most of them are autosomal recessive. That means when it is in the heterozygous condition, it's not going to cause any disease. Only when it is homozygous condition is going to cause the disease. Similarly, the Finnish population, you can see large number of diseases. Since these populations are very small and they have well studied, and they map the gene for all these diseases. But what we need to do is the one which I'll come a bit later, to hypothesis our finding that large number of populations are with population specific recessive mutations. We took this disease, progressive pseudo-rheumatoid dysplasia, which is caused by mutation in WSP3 gene. Again, the heterozygous condition, parents are absolutely fine. And you can see that the children who are born with the homozygous mutations have bone abnormality, particularly in the joints, as is clearly shown in the X-ray. They have a problem, short stature, and it's very, very painful problem. Again, to understand that, what we did, we sequenced these individuals who carry the mutation in heterozygous condition. We took, in fact, 30 patients. 6 of them are carrying the mutation in homozygous condition, and wanted to see whether they are from the common founder. We sequenced the flanking region of the mutations and found 5 out of 6 are having similar haplotype, right? Again, they say that they are non-consanguineous parents, but it tells that several generation back, they must have come from a common founder. So that is going to be a big challenge in country like India or South Asia. This is another example where you see that the consanguineous family, there's a gene called polymerase gamma, which is essential for mitochondrial DNA replications. And here you can see that both the parents are with heterozygous mutation. CT, as it's depicted here, the electropherogram. This is a wild type. This is a heterozygous condition. Both the parents are with heterozygous mutation, and unfortunately, 2 of their children are with homozygous mutation. Hence, we don't know the fate of this child, and this child was alive at the time when started the analysis, but after some time passed away. What is the reason for this particular mutation in terms of its association with mitochondrial DNA replication? You can clearly say this is a wild type 10 kb absolutely single band. The individual with the POLG mutation, one base change from C to T, which causes multiple mitochondrial DNA deletion, and you can see brain MRI showing the lesions. And these are some evidence. These are the ragged blue fiber, and these are COX negative fiber, indication of the mitochondrial disease. So to come to the point that the endogamy and population specific recessive disease, it's a consanguineous family. Even the non-consanguineous family, you see that these are from the southern part of India, where the consanguineous as well as the endogamy is highly practiced. Non-consanguineous, but they are endogamous marriage, where you see parents are heterozygous again with 41 and 38 years, but unfortunately both the children have died due to this mutation. So we don't know how many such cases or populations are existing in India. As you know that only very few populations who are living in the city or town can go to medical community. Otherwise, in the remote places where the endogamy is practicing, I'm not very sure that whether they get enough medical attention or these informations are being transferred to medical community or not. So, with that, so what we need to do is something like what Ashkenazi Jewish population practice. They have organization called Doreshim. This is a community genetic testing. They have a program where they go to school and collect the blood sample from the children, sequence them, put them in their database. When they grow old and come to the stage where they wanted to marry, the boy wanted to marry a girl, if he identified or she identified, what they do, they take out the sequence of both the individuals and assemble and see whether both of them carry the same mutation which are responsible for causing some of the disease or not. So that way, if one can actually avoid, as you know that avoiding is the best thing for any genetic diseases. So that way, in fact, they have eliminated a couple of diseases from their community. So this requires a lot of dedication, very long-term work. So in that line, we have now started looking at some of the population-specific diseases which are known. Now, there is one disease called Handikudu, where you can see that the individuals are with no start stature, bone abnormality with a lot of pains, and this is another this chronic disease called Udanam kidney disease, which is again the entire populations are suffering from that, and they are in the dialysis program. Similarly, we have large number of population specific disease. This is one aspect. Another one, based on our study where we identified a large number of population with a very high IBD score, although we don't have any clinical detail about the population, but we detected genetically there's a possibility of having population specific mutation which may be responsible for population specific disease. So we are analyzing them, sequencing either whole genome or exome, identifying the mutation, then going back to the community, and once we identify the mutation, we'll screen the entire population and provide enough counseling and other support. So that's our new program which we initiated when I was in CCMB. Recently I joined CDFD, which the work is still continuing. So with that, I would like to thank all those who have contributed to the study, right from Dr. to many of my students and colleagues. And I have wonderful collaborators. I recollect what Vice Chancellor was mentioning, that we need to have a very good collaboration, because one individual or one lab or one department cannot do such a massive work. I once again tells that the amount of collaboration which I have for the last 2.5 decades is the one which in fact helped in bringing to this level. And with that, I also like to thank the funding agencies like CSIR, UK India Education Research Initiative Grant, DBT, DST, and Medical Research Council at London. Thank you very much.
Thank you, Dr. Thangaraj, for a very elaborative and very informative lecture. We all have listened to you very fervently. And if you permit me, I would like to take 1 or 2 questions from the students and scholars
Sure.
who are there on our YouTube platform. So, sir, the first question is by Akanksha Sharma, our MSc student. She wants to know, do human centromeres have signatures of genome sequence? Are the sequences of all human centromeres different or same?
The centromeres are in fact having full of repetitive sequence. It's highly with the heterochromatin. So some of the sequence initially was done is lack of that region, because very difficult to sequence the region which has a repetitive sequence. Of course, now there are different technology which very long read is able to do that.
Okay, thank you, Dr. Tangaraj. Next question is by Srishti Valecha. She wants to know how Neanderthals lost their Y chromosome in the trajectory of human evolution.
No, they have not lost their Y chromosome. I don't know who said, but there is a prediction that the Y chromosome may we may lose the Y chromosome, but I don't think that will happen.
Actually, it was there that some of the males, they vanished gradually in the trajectory of their conversion in human evolution.
Yeah, of course, it has been shown that the Y chromosome in fact evolved from the X chromosomes. It has lost most of the part, accumulated a lot of heterochromatic region and sex determining region. But even today, if you see, this large number of individual might have a shorter Y chromosome where there is a deletion of heterochromatic region. But the SRY, one of the genes which are responsible for making individual as male. In addition to that, there are few more androgen receptor gene and other things. But SRY, which is present on the Y chromosome, which is a master regulatory gene for maleness, should be there for make individual as a individual. But even lot of heterochromatic region goes away from the Y chromosome. The Y chromosome may exist as a very tiny Y chromosome, or after several millions of years, it might translocate on some other chromosome which nobody can predict, right? But there will be a, if not Y chromosome, that there will be some other chromosome which will carry the sex determining region.
Thank you, Dr. Thangaraj, once again for your knowledgeable presentation. There are so many other questions, but I must not take them because of the time constraints. Now, next speaker today is Dr. Vasudha Sambayal, Professor Guru Nanak Dev University, Amritsar. Although we all know her very well because of her polite nature, humble behavior, and special knack of socializing. But let me formally introduce Dr. Vasudha to all of you. Professor Vasudha did her master's in human biology and doctorate in human cytogenetics in the same very department. Then after completing her doctorate, she joined Department of Human Genetics, Guru Nanak Dev University, Amritsar, as lecturer in 1998. Professor Vasudha became head of the department in 2011, and then onwards, she has been either member or chairperson in several prestigious professional bodies and committees. Her area of research is human cytogenetics and cancer cytogenetics. Besides supervising 8 PhD students, she was awarded 8 research projects from different funding agencies such as UGC, DBT, DST. Dr. Vasudha is recipient of ISTA Young Scientist Award and Rotary Appreciation Award on Teachers' Day. In reference to her impressive research publications, she is one of the finest human cytogeneticists that we have in North India. Now I request Professor Vasudha to please share her valuable research.
Thank you, Dr. Puneet. These are words for introducing me. I think you give me more credit than I am actually capable of. And I am very thankful to the Department of Human Genetics, the Honorable Vice Chancellor, Punjabi University, Patiala, to allow me to present my work here. It is really humbling to be presenting in my alma mater at its 50th Golden Jubilee webinar. I was a student long back in the department when Dr. Sidhu was referring about our 25th Jubilee celebration. That was a Silver Jubilee celebration. I was a PhD scholar at that time. And from there on till now, there are many things that I still carry of the department with me. There is a lot that I learned from my seniors, and there is a lot that I still continue learning from them. Today, listening to Dr. Sidhu, as the Honorable Vice Chancellor also mentioned, it was a very great thing. And also, I am going to talk after Dr. Thangaraj. It is intimidating to talk after Dr. Thangaraj. So the illuminative talk that he has given, the extent of work he has presented in summarizing it and putting it across so beautifully, I'll be talking about a little aspect of that. So I was talking about population related work and population genetics, and in context of that, the disease risk which are specific to certain populations. I'm going to present a work which is dealing with the risk for cancers in a population that was largely from what is known as Punjab now. Okay, just give me a minute to put up the presentation. Please just confirm whether the slides are visible. Dr. Puneet, please confirm whether you can see the slides.
I request Professor Thangaraj to please mute and stop presenting if he is presenting still now. Okay, Vazai, you may present, but your presentations is not visible here.
Just I'll do it again. Actually, okay, now it will be there. Please check now whether the slides are visible.
Yeah, visible.
Okay, so shall I start?
Yes, ma'am.
Okay. So I'll be presenting on my work, which is starting from cytogenetics and then going on to the molecular aspect of it. As Dr. Puneet mentioned earlier, I am a cytogeneticist by training. My training was in Punjabi University, Patiala. I learned a lot from my teachers there, and taking it on as I came up to this place, that is Amritsar. What I am presenting now has its genesis in the work I did with Dr. Sidbeet and under the guidance of Dr. Bansal in Punjabi University, Patiala, almost about 20 years back. And the work that is being presented now is a summary of what we have done in the past about 10 years. Okay. Now, this starts with chromosomal instability. The idea is that in tumor samples, when we look at the tumor samples, we see a lot of cytogenetic instability. And this is also known that when you have this instability, this leads to disruptions of many of the signaling cascades. So the idea was, can the cytogenetic instability be used to decipher the genetic pathways which are involved in carcinogenesis? We have a lot of literature which is available for this in context of what we do from tumor sample. Our idea was, could we do it from a non-target tissue? Okay, so this is what we were looking for. We were looking for breast cancer in journal to look for the genes which are involved in breast cancer, specifically in our population. Since in Punjab, we have a fairly high incidence of cancer, and among them, breast cancer in females is the highest in frequency. So we were looking at the chromosomal regions which were recurring in aberrations in the non-target tissue, that is peripheral blood lymphocytes, and could we use that information to find which genes were involved in breast cancer in the samples that we were talking of from our population. This population was largely that was from Punjab. Okay. And then whether we should look for the polymorphic risk alleles in these genes, and could that be used to predict either breast cancer risk or could it be used to predict the response to therapies. Okay. So for this, about 400 subjects had been selected, and after collecting their history and the blood samples, cytogenetic analysis was done. After cytogenetic analysis, the chromosomal loci which were involved in recurrent aberrations were checked up in this. This data was then used for in silico analysis, and this was also followed by the molecular analysis. Now, when we looked at our sample, we found that majority of our sample, more than 96%, were female only. The frequency of males was pretty less. Majority of these subjects were more than 40 years of age, and also that when we looked at the female sample, almost more than 50% of these were postmenopausal samples. Whereas with reference to the family history, only about 37 patients reported a positive family history for cancer in general, not specifically for breast cancer, and about 80% of them said that they did not have a family history, at least for a 3 generation, what they could recall of. So it appeared that majority of these were sporadic cancers. Okay. And most of these patients had reported in the hospital at the second stage of cancer. Our sampling was from Guru Ramdas Institute of Medical Sciences in Amritsar, where our collaborating physicians and surgeons were there. Now, in that sample, on basis of their histological type, almost 90% was infiltrating ductal carcinomas. Okay. So when we looked at the cytogenetic aberrations, we found that the cancer patients were having a lot of anomalies. We had both the chromosome type and chromatid type anomalies. There were structural aberrations. There were numerical aberrations. There was more of a loss of chromosomes than a gain of chromosomes, which was observed in these patients as compared to what was seen in case of controls. So what we did was, for each patient, we took about 100 clear metaphases. We classified the metaphases in context of those which were showing just numerical anomalies, or those which were showing only structural type of anomalies, or those which were showing both these type of anomalies. They had structural as well as numerical anomalies. We counted all of these metaphases, categorized them accordingly, and then we took out the mean of all these values. But still we found that the number of aberrant metaphases were much more in patient samples as compared to what you had in case of controls. Okay. And for various categories also, the patients were having more aberrations. Now then what we did was we compared a stage-wise distribution. We found that the aberrations were still higher compared even in a stage 1 patient compared to what was that in case of controls. So the next point was to look for which of the chromosomes were showing these changes. So what we did was we took each category of aberration that we were getting, categorized them in context of which particular chromosome was involved in that, and then again compared the patients as well as controls. Okay. And again we found that in case of patients, there were more aberrations where we found a stickiness of the chromosomes. Then we also found there were interchanges which were more, and also structural aberrations which were comparatively more in case of patients than controls, and some specific chromosomes appeared to be involved in this. Okay. Now, using these aspects, we went on to using the bioinformatics tool. The in silico analysis was done, in which the chromosomal loci which appear to be involved in the recurrent aberrations in the cultured lymphocytic tissues of the breast cancer patients were identified. Then from using these two databases, that is Atlas of Genetics, Cytogenetics in Oncology and Hematology, and the Gen Atlas database, the genes which were present on these loci were retrieved. Okay. We got a list of genes at all these particular regions. Now, using this information as a background, the next part of it was that using the rectum F1 method, the functional interaction networks were generated. Now, this helped in identifying that which genes were comparatively in a higher number in our sample with reference to what we were getting. And this is what came out of it. It turned out that the pathway enrichment, when it was done for invasive ductal breast carcinoma, since most of our sample was infiltrating ductal carcinoma type, we found that these 6 genes were involved in infiltrating ductal carcinomas: EP53, HIF1 alpha, AKT1, EP300, PIK3CA, and SMAT4. Okay. Then another aspect which came out of it was when the analysis was done for genes which were not linked together in the network construction again, it turned out that these about 6-7 genes, including HTAG, NCOA1, NLR34, COLA1, RARA, WTR1, and BRCA1, were enriched in the RNA polymerase II transcription pathway. None of this was not really new. RNA polymerase II transcription pathway has been known to be involved in tumorigenesis. And also, p53 is a known tumor suppressor. HIF1 alpha is a gene which has been known to be involved in cancer biology. Similarly, many of these others are also known to be involved in various cancers. The only thing was how then this information could be used for the breast cancer studies. So what we did was we took up the first two, that is p53 and hypoxia and HIF alpha, because hypoxia is a known factor that leads to aggressive tumor phenotypes, as well as it has been known to be one of the components responsible for therapy resistance. And whenever you have a hypoxic tumor, the chances of that becoming more aggressive is comparatively much higher. And nowadays, hypoxia is one of the factors which is also being targeted for therapy. Okay. Related to hypoxia, we found that there is the second aspect, that is angiogenesis, and of course it triggers genome instability as well. So the hypoxia cellular response has been found to be mediated by 3 of these factors: HIF1, 2, and HIF3. And when we were talking about p53 and HIF, we found that activation of HIF1 and accumulation of p53 are very typical in hypoxic tumors. So we concentrated on these two genes. And what came out of it also was that p53 has been involved in the process in which we say there is a ubiquitination and then the proteasomal degradation of HIF1 alpha subunit of HIF1, okay, in conditions of normoxia. Another thing what has been known is whenever there is conditions of persistent hypoxia, that means less than 10 millimeter Hg of oxygen levels that you have, at that point HIF1 alpha is going to translocate, combine on with HIF1 beta, and then this product is going to join on to the hypoxia response elements of multiple genes, and among them one major gene was the vascular endothelial growth factor, which was responsible for angiogenesis. And in tumor samples, angiogenesis is another aspect which has been found to be related to the spread of the tumor or the cancer. So we targeted this, that we can look for HIF, we can look for p53, and third we should be looking at the vascular endothelial growth factor, because that was related to the angiogenic switch that you have during the process of tumorigenesis. Now, then in VEGF, as we looked at it, we found that the vascular endothelial growth factor is going to act on the cell using another protein which is known as the receptor, and whenever there is hypoxic stress in tumor samples, the transcription of hypoxia inducible factor 1A is activated. This also promotes increased VEGF expression, and whatever is the VEGF there, this is going to bind on to VEGF-R, which is on the surface of endothelial cells, and this has an effect on angiogenesis. Apart from that, it will be also increasing the MMP expression in tumor cells. So that was the fourth one that we targeted, that is VEGF-R. Now, using this, we looked at the polymorphic variants for these 4 genes, that is TP53, HIF1 alpha, VEGF, and VEGF-R1, In all, we selected almost about 22 polymorphisms. Some of these were selected with in context of their earlier reports in other cancers. Some of them were selected on basis of their relation with the expression of the gene or the transcription of the gene. And in all, we selected 5 for P53, 3 for HIF, almost 13 for VEGF, and 1 for the receptor. For many of these, there was not much information available from various Indian population, and specifically from North India and Punjab. There was not much information available. So that was another reason to find out the population specific information also for these. Okay. Now, using PCR, RFLP, or the ARMS PCR method, these polymorphisms were analyzed. For HIF, we had selected these three, the S285, the P582S, and A588T. It turned out that these two, this one and this one, S28Y and A588T, both of these turned out to be wild type, completely wild type in our population. Both in patients and controls, we did not see any variant in this. Whereas for the 582S, that is C1772T, as you talk of, we were able to see that there were again more of the homozygous wild type, and there were some individuals who were heterozygous for this. Association analysis indicated there was no significant association with any particular model. Okay. For P53, 5 had been selected. Out of these 5, again, two of them R213R and P47S. This one and this one. These two again turned out to be wild type, completely wild type in case of our population, and there were all the patients and controls had wild type genotype for these two polymorphisms. Whereas for R72P, this was a polymorphism which had been selected because it was supposed to be having a relation with the response to chemotherapy as well. In this, the heterozygous genotype was comparatively higher compared to wild type or the homozygous variant. But again, association analysis did not show any significant association with any particular genetic model. Okay. For the next one, which was an intronic polymorphism, again, the association analysis did not show any significant association with any genetic model. We had more of the wild type compared to the variants, and both patients and controls had nearly similar values for the heterozygous individuals as well. Okay. For the last one, that was 13494 G2A. Again, in this case, the association analysis did not find any significant association, whereas again we had the homozygous wild type compared to in a higher frequency compared to the other categories. Now, this indicated that when we talk of individual polymorphisms for P53, which is a known tumor suppressor, we were not seeing any definitive association with our breast cancer patients. Okay. Then we looked at the interaction between P53 polymorphisms, and it found that this combination where we had for C1772, where we had for R72P for PIN3 as well as for 13494 G2A, this was comparatively higher in patients compared to controls, but the difference were not statistically significant. Okay. So what came out of P53 was we were seeing a higher frequency of the RP genotype for R72P. There was more common A1 allele for, that is a wild type allele for PIN3. Again, GG, that is a wild type, was more frequent in breast cancer patients. And the interaction indicated that the intronic polymorphisms were in a higher frequency in compared to in patients compared to controls, whereas RP A1A1 GG was in an increased frequency in breast cancer patients. Okay. Then we looked at the interaction of P53 with HIF1 alpha, and in this we found that there were these two combinations among the many that were generated. This was higher in patients compared to controls, whereas this particular combination was comparatively lower in patients than it was in controls. Okay. Still, the differences were not much statistically significant. Okay. Then we looked at the vascular endothelial growth factor, and in that we had done multiple polymorphisms. And when we looked at these polymorphisms, we found that for 2578C2A, this indicated an increased risk with the AA genotype. Okay. Again, looking at the next one, that is 2549 insertion deletion polymorphisms. Again, this showed an increased breast cancer risk with a significant association with the II genotype as well as the ILE. Okay. Then for 460 T2C polymorphisms, there was an increased breast cancer risk which was seen with the CC genotype as well as the C allele. Okay, and the genetic model was indicating increased risk with the codominant as well as the recessive genetic models. Okay. For 405 C2G, again we had a significant association for the GG genotype as well as G allele, showing a significantly increased risk for codominant as well as for the recessive genetic model. Okay. Now, in vascular endothelial growth factor, some of the polymorphisms were done by direct sequencing, and in that case, it again showed that 152G2A was showing an increased risk with the breast cancer, with the AA genotype associated with breast cancer, and also the recessive genetic model and codominant model significantly associated with increased risk of breast cancer. Again, for 116G2A, it was the AA genotype as well as the A allele. This was showing an association with increased risk of breast cancer. Okay. Then there were some of these which came out, which were showing a comparatively reduced risk of breast cancer. So for VEGF 165C2T, the CT genotype as well as the T allele was associated with a reduced risk of breast cancer. We did not see any TT individual in the population. Okay. For VEGF 141A to C, the AC genotype and the C allele was showing a decreased risk with breast cancer. Again, in this group also, we did not see any individual with the CC, that is the variant genotype. Okay. So what came out of VEGF was that the combination of the 7 promoter polymorphisms, these were associated with an increased breast cancer risk. Also, the ALE, the ILE, the C allele, G allele, C allele, and the C allele combination was associated with an increased breast cancer risk. Whereas the C allele, D allele, T allele, G allele, C allele, and C allele combination of these polymorphisms appear to be associated with a comparatively reduced risk. Okay. So the VEGF haplotypes gave us two types of information. Some of them appear to be associated with a comparatively increased risk of breast cancer, whereas some other combinations appear to be associated with a reduced risk of breast cancer. Okay. Now, out of this, 2578 and 2549, they were in linkage disequilibrium. Also, 2578 and 460 T2C were also in linkage disequilibrium, and same was for these two also. Okay. So when we looked at the VEGF diplotypes, it turned out that some of these diplotypes were present in a much higher frequency in breast cancer patients as compared to what we saw in controls. Okay. And with reference to this, when we looked at the other genotypes, we found that some of these genotypes were present in a comparatively lower frequency in patients as compared to controls. So these indicated, these combinations indicated a higher risk associated with breast cancer for these diplotypes. So then we looked at all the four genes and their polymorphisms, that is HIF1 alpha polymorphism, the p53 polymorphisms, the VEGF, as well as the VEGF-R1 polymorphisms. And we found that in this, some of them were coming up in a strong linkage disequilibrium, whereas we have the p53 polymorphisms, that is PIN3 as well as 13494 intronic polymorphisms. And for VEGF also we had the 2549 as well as 460T to C, 2578 as well as 2549 insertion deletion, which were showing a strong linkage disequilibrium. So on basis of this, when we looked at the complete haplotype analysis for these categories, it turned out that this particular combination was associated with a comparatively increased risk, whereas we had this particular combination which appeared to be associated with comparatively reduced risk for breast cancer. Okay. So when we looked at these all four genes and their polymorphisms, we were able to find out a specific combinations which appear to be indicating that these were associated with increased risk in our population. So to summarize, what we found was when we looked at the genes individually, that is when we looked at just HIF1 alpha, the T allele appeared to be increased in patients compared to what we had in controls, but the differences were non-significant. But when we subcategorized the population and we looked at the menopausal, premenopausal and the postmenopausal patient, the T allele appeared to be significantly associated with breast cancer risk in premenopausal patients only. When we looked at only TP53, we found that the proallele of R72P polymorphism, the A2A2 genotype of PIN3, and the AA genotype of R13494G2A polymorphisms, these appear to be in higher frequency in patients than controls, but the differences were not statistically significant. And also one genotype of 53, this was in higher frequency in patients. Another interesting thing which came out of this was then we compared all these with what we were getting from the chromosomal aberrations, and in some of these patients had also been followed up post therapy also. So it turned out that the patients who were having these combinations, PP for R72P and A2A2 for PIN3, also those who were having the RR-GG combinations for R13494 and R72P, and individuals who were having this genotype for HIF1 alpha, for R72P, and for the PIN3 as well as for 13494. These had increased chromosomal damage compared to other patients, and they had a lot of numerical as well as structural anomalies in their samples. So it somehow indicated that these were also related to the overall amount of the overall function of the protein and the way it was dealing with the chromosomal damage which was occurring in a non-target tissue, probably because of the physiological stress which was generated by the presence of a cancer in the body. Okay. For VEGF, it turned out there was a significant association with increased breast cancer risk of the AA genotype for 2578, for the II genotype for 2549, for CC genotype of the 460C, the GG genotype for 405, AA genotype for 152, and AA genotype of 116. These were all associated with increased breast cancer risk, whereas there was a significant association with the reduced breast cancer risk of two of these alleles, 141C and 165 TLA. Okay. Now, on basis of this, when we looked at all of them together, we found that this particular combination of HIF, P53, VEGF, and VEGF-R, this was associated with increased breast cancer risk. Compared to this particular combination of HIF, P53, VEGF, and VEGF-R, this was associated with decreased breast cancer risk. So this is all that this is a work which came out from what we started from the cytogenetic anomalies in a non-target tissue. We summarized that, we found out that this was indicating some specific genes which were involved in our breast cancer sample, and out of that we took up the first two genes and we ended up with this. So this has given us information in context of hypoxia as well as the angiogenetic pathway, which can then be used for further estimating risk as well as for therapy related things. So for today, that is all that I am presenting, which was the basic kind of a research on two aspects of genetics, the cytogenetics as well as the molecular biology. So these are the conclusions that came out of it, that the recurrent aberrations involving specific chromosomes can indicate the loci which are associated with cancer. This can be used to identify genes which may be critical in carcinogenesis in that particular group, and therefore the chromosomal instability analysis in a non-target tissue can also be used for it. and then polymorphic alleles of these genes can then predict breast cancer risk. The work that I have presented today has been the work of Dr. Rohi, Dr. Sarika, and Dr. Akin Kaur, and under the able guidance of my colleague, Dr. Kamlesh Kuleria. And all these samples were from our clinical collaborators from Sri Guru Ramdas Institute of Medical Sciences, with the projects which had been supported by DST, DBT, and UGC. And the work that I have summarized today was presented as these publications in the past few years. Thank you all for your patience.
Thank you, Dr. Vasta, for shedding light on this dreadful disease, which is highly prevalent in our population. And this disease is also having very severe clinical connotations. I was reading the other day that the highest prevalence in North India is, I think, in Chandigarh. Is it so?
There are reports which indicate that, but even in certain areas of Punjab, we have very high frequency.
Okay.
Areas around Mansa, Batinda, and others have very high frequency for cancers, much higher than other parts of Punjab.
Okay. Vasaji, now may I take the liberty to take one or two questions from our attendees?
Sure.
First question is by Ruben. She wants to know, are there any P53 targeted anti-cancer agents currently available or under development in India?
There are certain agents which are being targeted, but I am not sure whether they are being developed in India. There are multiple reports from other countries which are developing 53 targeted therapies and drugs. From India, if any company is developing, I'm sorry, I would not be able to identify that. Dr. Puneet, I could not hear you. Your mic is off.
Sorry, Vasaji. The second question is by Nitin, our research scholar. Is there any degradation seen in P53 because of COVID-19 infection?
Degradation in which context?
P53 protein, he wanted to know, I think.
Okay. Then I am not.
Interaction of this P53 or COVID-19 infection, actually.
Can you repeat the question again? There was a break of voice.
Is there any degradation seen in P53 because of COVID-19 infection?
I don't. I would not be able to comment on that.
Because there are some coming that if there are comorbidities along with this COVID-19 infection, that then it becomes aggressive. Actually, he wanted to know maybe that during COVID-19 infection, is there any interaction of this infection with P53, etc. So that's maybe.
There are some reports which are indicating in context of the role of 53 in the damage which is being caused even to lung cells. But whether that is relating to degradation in the protein, that I am not sure of as of now. There is still, I think, more work needed on that to be sure of that. So I would not be able to give a clear answer on it.
Vasaji, one question from Kamaljeet Kaur. She's also a research scholar. Can BRCA1 and BRCA2 carrier status affect the association of these genes? These 4 genes that you picked?
Well, BRCA1, some individuals who have BRCA1 mutations or maybe having BRCA2 mutations do have, then subsequently tend to have more variants that we see for 53 as well. Okay. And there are reports for that. And similarly, we have for AKT and SMAT4 also, which are linked to it to some extent. So yes, because these are all part of like BRCA1 is part of that cascade where it is dealing with DNA repair and many of these genes are interacting with each other, so they do have an effect.
Okay, thank you very much, Vasaji, once again.
Thank you.
Now, the next speaker, now eminent personality that we have with us today is Dr. Sarbjeet Mastana. Dr. Sarbjeet Mastana is senior lecturer in human genetics in the School of Sport, Exercise and Health Sciences at Loughborough University. An alumnus of Punjab University, Chandigarh, Dr. Mastana did MPhil from Cambridge University and PhD from University of Newcastle upon Tyne, UK. He has been teaching for the last 25 years in the areas of human genomics, functional genomics, human evolution, forensic DNA analysis. Dr. Mastana's research expertise is in the field of population and disease genomics. Currently, he is focusing on genetic and epigenetic implications of several complex disorders such as diabetes, heart diseases, hypertension, osteoporosis, and rheumatoid arthritis. While guiding several projects in graduate, postgraduate, and doctorate programs, Dr. Mastana has around 100 publications in very prestigious journals like Nature Review Immunology, Current Biology, International Journal of Cardiology, BBA, and many more. He is editor-in-chief of Internet Journal of Forensic Sciences, editorial board member of International Journal of Human Genetics, and Journal of Biological Anthropology. Now I request Dr. Sarbjeet Mastana to please share his valuable research.
Thank you, Dr. Puneet, and thank you, Professor Guman, Vice Chancellor, Punjabi University, Dr. Rajinder Kaur, and other colleagues. And thank you to Professor Thangaraj and Professor Vasudha. And it is a Herculean task for me to follow them in terms of presenting what I'm doing here in Loughborough University. And we will have a kind of discussion about my journey in human genomics, and it has contributing factors from Professor Sidhu also. Professor Sidhu was our examiner at many examinations in Punjab University in between 1979 and 1984, and I contribute my growth also to Professor Sidhu in that process, and I have met him many times in that process when he visited us in Newcastle upon Tyne, as well as in London. My talk is going to be about my journey and potentials for future activities in that process. Now, if Dr. Vasudha, if you stop presenting, then I can start presenting.
Hanji Dr. Sahab, you can go ahead.
No, but I think Dr. Vasudha is presenting still. Okay, now I'll present now.
Okay.
So as I said, I mean, my journey is via Punjab University, Chandigarh to Loughborough University via Cambridge and Newcastle upon Tyne. So I'm going to talk about population genomics in health and disease, hope, hype, and potentials. And that basically is following up from Professor Thangaraj's excellent presentation, followed by an excellent presentation by Dr. Vasudha in terms of that cytogenetics plays a significant role in our understanding of personalized medicine or personalized genomics. So in terms of outline for my presentation, I'm going to focus on why do I care about studying genetic variation? What is the benefits? What are the limitations? And what is personalized medicine? And what are the implications of that? And why Indian populations have potential for achieving that genomic medicine or personalized medicine. And we will be addressing some of those what you call activities by carrying out a range of studies in our field of genomics and epigenomics, and looking at sport and exercise as a health outcome in that process. Dr. Puneet has already introduced me, so I will probably skip this slide. But in terms of my pedigree, I'm from Punjab University, Chandigarh, and then from Chandigarh I went to Cambridge University. From Cambridge University I went to Newcastle upon Tyne, and for last 15 to 20 years I've been focusing on human genetic variation studies in human populations, diseases, and looking at public health implications of that information. So in terms of our work, we are mostly interested in what the health outcomes are going to be, whether we will be healthy or whether we will be ill. And health outcomes are dependent on a range of activities in our portfolio or in our biology. One of them is called genomic variation. Genomic variation is basically the variation in our DNA. Now, DNA is present in our cells, and those cells have a lot of variation in that process of, and we can study that variation, but that variation on its own is not the solely responsible for genomic activities, etc. So we also need to study our environment. And environment is everything under the sun. So environment contains for our physical activity, our food habits, our stress levels, our smoking, drinking, all those kind of activities are under the umbrella of environment. Whether we are able to afford good quality foods, that is also part of that environment in that process. So in terms of how our health outcomes will depend upon are dependent on genomic variation, dependent on environment, and also dependent on word called epigenetics. And epigenetics is basically beyond genetics or above genetics. So that's a very major field of research activity which is happening now, which is contributing towards our understanding of disease and understanding of how the disease happens in different populations, different contexts, and this is what and how we can modulate those diseases, how we can change our epigenetics or epigenomics in that process. These are some of the cartoons from various news magazines, etc. And basically, they're showing that there is a lot of interest in genetic research. There's a lot of activities going on in thinking about the genetic research is responsible for medicine, is responsible for our personalized medicine, as well as our social aspects. For example, if in US, if you don't have a gene test, you may lose insurance. If you can be labeled by virtue of carrying different combination of chromosomes or different combination of polymorphisms as born bad in terms of criminal activities, or you may start believing in that your behavior or your personal behavior is dependent on your genes. Now, keeping all these things in mind, there is a lot of awareness of genomics or genetics in general populations. People are more aware of it. And there are a number of companies which are offering genomic activities or genomic tests which are available to us now. For example, these are two advertisements basically showing you that inside genomics, your future, your fate, when it is your health, how much do you really want to know? So there is implication for individuals, implications for families, implications for populations and society as such. The future of your future of health is in your DNA. A range of these what you call testing regimes are available from many companies now, and they are easy to analyze. They're easy to collect. Most of the time, we are focusing on, for example, cheek swab samples. Cheek swab samples or cheek saliva samples are sent to these companies, and they will analyze. For example, 23andMe will analyze almost everything under the sun using their what you call SNP genotyping, and then there are specific companies which are ordering, for example, analysis for specific pharmacogenomics, specific skin exercises or ancestry or other aspects. And these things have increased our awareness of that genetic variation needs to be studied. And there is large amount of genetic variation present in different populations or different groups of individuals. And why we need to bother about studying genetic variation? And basically this cartoon indicates or shows that if we don't have genetic variation, then we will look, or all of us will look same, and that will not be a very useful thing. We will all have the same genetic risks, we will have same disease risks, etc. So we need to kind of study genetic variation. Genetic variation is basis for human evolution, and Dr. Thangaraja clearly highlighted that human evolution is based on potential for genetic variation, and in other words, genetic variation leads to human evolution, and human evolution leads to genetic variation in different population groups in that process. So why do we care about genetic variation? In terms of our main focus, we are interested in how it impacts on our health and disease at both levels, population level as well as individual level. So population level, because we need to think about what tests should be available. At individual level, we need to think about treatments. So right drug at the right dose at the right time. And that is basically the main premises of what we call personalized medicine. Now, personalized medicine is a beautiful word, and everybody wants to do the personalized medicine, but it is very, very challenging and very difficult at this stage in the process. So let's look at why we think personalized medicine will be helpful in future, or what is the need for personalized medicine. At the moment, what we have is a trial and error method of medical prescription. So everybody prescribes the medicines based on their understanding of pharmacology, based on their understanding of what that drug is supposed to do. So current practice is one size fits all, and we call it trial and error method of prescribing. And trial and error method of prescribing, it has limitations because it is not suitable for everyone. Everyone doesn't get benefit. We can also call it imprecision medicine. Now on this cartoon, you have for every blue person, there are 3 to 24 people who do not get any benefit. So there is a lot of wastage in that process. Here, for example, arthritis drug, Humira, is only suitable for 25% of the people. Here it is less than 20%. Here it is less than 20%, 20%. This is a high cholesterol drug. This is heartburn drug, schizophrenia drug. So drugs are not suitable for a large number of individuals because they may have different combinations of genes, different combinations of polymorphisms, which do not allow them to be metabolized properly, or they don't give any major benefits. Similar kind of example, here we have again lot of hypertension drugs are not suitable for 10 to 30% of the population they're prescribed to. Asthma drugs, 40 to 70% of the individuals do not get any benefits, but they can continue to accumulate what we call side effects, and they can continue to accumulate uh, uh, chemical buildup of those chemicals, and in other words, they do more damage than actually helping individuals in that process. So the benefit or the purpose of personalized medicine is the right treatment for the right person at the right time. And to achieve this, we need to know about genomics of those individuals. We need to know the genetic combination of those individuals so that we can understand which medicine is right for which group of individuals and how those can be prescribed. So why we are not getting there? So this is a typical definition of personalized medicine. So personalized medicine will enable us risk assessment, diagnosis, prevention, and therapy, which is unique to the individual and will enhance the quality of life and public health. So why is it not offered in clinics? The main issue we have is that we have very limited genomic information. We have very limited genetic information from individuals. We have some genetic information, we have some clinical information, but we also have very limited knowledge of how the person will develop the disease, its response to the treatment, course of the disease, etc. So we need to have more information about these parameters. And a lot of activities are going on, a lot of research is going on, where we are contributing towards this process. The problem we have is that our health outcomes or any outcome in that process is not dependent on single gene or single environmental factor. Here we have an example of a very simplistic example, in fact, that many genes are involved with different effect sizes, and many clinical parameters or demographic parameters are involved, again with different what you call sizes in that process. So in terms of main contributors to health and disease are variable between diseases. Dr. Thangar has also highlighted, and Dr. Vasuda also highlighted, that there are many genes which are involved into, for example, breast cancer etiology, and many genes are involved into cardiomyopathies in that process. So what we have been doing for last 2 decades or so is we have been carrying out range of experiments, and those range of experiments are giving us some indicators or some understanding, and those tools are being used in terms of developing the picture. And developing the picture is basically that we need to carry out many thousands of experiments. Those thousands of experiments will lead us to develop a picture, and that picture is still not very clear. And to develop a clear picture, we need to still carry on doing those activities in a concentrated manner so that we are able to do those contributions towards our understanding of the disease, understanding of the health outcome. So where are we now? We have identified millions of SNPs, or single nucleotide polymorphisms. But majority of these associations have been observed in European populations. Most of these GWAS studies or genome-wide association studies are limited to European populations. Also, there is a limitation that majority of these Jiva studies only explain small fraction of variability. And what are the reasons why these are not same in every population or every group of populations one could study? Maybe there are local issues, local uh, population dynamics, which are important contributions towards understanding of that disease. So, what are the implications of lack of diversity in genetic studies? And what are the potentials for Indian populations to contribute? So we have already highlighted that we have a lot of results from GWAS, and they do allow us to think about function, think about pharmacogenomics, think about risk prediction, and think about the therapy or drug developments in that process. But we still have issues in terms of that their contribution in clinics is still limited to some extent. Why that is the reason? The reason is that if you look at genomic research databases, most studies are done on European cohorts. Dr. Thangaraj mentioned number of Indian cohorts, but still in 2017, most of the focus was on European cohorts, and a very limited number of studies were from Indian subcontinent in that process. So lack of diversity in genomic database is a barrier to translating precision medicine into research, precision medicine into practice. So that's a kind of a lead article in terms of precision medicine in 2018. Basically, what it is saying is that we need to study more populations, and we need to understand more populations so that we can have a good, clear idea where those errors are, where those polymorphisms are, and how we can use them in using for precision medicine. So, in terms of why we are talking about that, if we don't study populations, we don't study different populations or genetically diverse population, that we will have limitations to deciding on biomarkers. So lack of genetic research in different populations will have impact on biomarkers, and those biomarkers will be we may be missing them, we may be making pharmacogenomic errors, we may not be considering efficacy of those therapies, we may be looking at conflict of evidence risk, and we may be basically missing the diagnosis. So these studies need to be conducted in situ in the populations of interest in those countries where there is missing element or there is a limited number of studies which have been carried out. Now, India in the past, and India in what you call has a very good anthropological studies conducted, and range of those studies have contributed to our understanding of genetic variation. And one of the activities we have been doing for last few decades is focusing on genomic and genetic variation in different populations of Indian subcontinent, either in my previous institutes as well as in Loughborough University. So if we want to have success in personalized medicine or personalized genomics, we need to understand genetics and genomics of diverse populations. And India is a very good example of that process. And because, as Dr. Thangaraj showed, there is a lot of genetic variation, and there is a lot of unity also. And a number of studies have now shown, and we have also shown that in our many research papers, that we have a range of those activities in that process at many genetic levels, whether you conduct it on standard polymorphisms, SNPs, microsatellites, or VNTRs, pick up any of those ones in that process. So this is similar to Dr. Thangaraj's slide in that process, that we have linguistic variation, we have uh, 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 geographical variation, we have climatic variation, and our populations have what we call endogamy as a big player in process of shaping our genomic variation in that process. Also, we have what we call very high level of type 2 diabetes and heart disease, and those need to be examined at genomic level as well as at epigenomic level. Similar to Dr. Thangaraj's, there have been studies on Indian populations, and those basically, here is a principal component analysis, which basically shows that Indian populations or Indian subcontinent population make their own cluster compared to, for example, European or East Asian populations, and those clusters are unique to Indian populations in that process that we do have a lot of individual level genetic variation or individual population level genetic variation, and but overall we make a loose cluster of human populations. We have, whereas in European population the cluster is much smaller, East Asian population also the cluster is much smaller, whereas in Indian population or Indian subcontinent populations the cluster is much bigger, and these are based on limited number of samples. Dr. Thangaraja also showed this ANI ASI mixture, and from their lab basically take home messages that our populations are relatively young. That means there are potentials for conducting epigenomic research as well as genomic research, because they have endogamy, they have social practices, they have uh, 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 cultural practices which are unique to our population groups. Now, this is the recent outcome from, as I said, I mean, original studies were based on SNP analysis. Those PCA and other analyses were based on SNP analysis. This indigenome is a CSIR outcome or recent outcome from CSIR. In terms of indigenome, they have analyzed around about 1,000 individuals based on high level of sequencing, average level of sequencing around 30 times, but they have very limited sampling. For example, they have only 16 individuals from Punjab, only 19 individuals from Andhra Pradesh, and haven't paid attention to, for example, any consideration of tribal, caste, and other classifications in that process. But take-home message here is that these 18 million SNPs are unique to Indian populations in this limited sample of 1,029 individuals. So that means there are large number of possibilities that when we survey mode genomic variation, there are going to be better outcomes in terms of our considerations for personalized medicine. So we need to study more populations, more clearly defined populations, more clearly surveyed populations, rather than simply taking samples which are convenience samples. So anthropologically, we need to work on those groups, work with anthropologists, work with population population geneticists in that process. So what we have in terms of Indian genome, we have genomic unity in diversity. So we have extensive variation even with a small sample size. Dr. Thangaraja also talked about unique and shared heritage we have. Unique set of SNPs we have, which may have implications for disease associations. Dr. Thangaraja as well as Dr. Vasuda showed that disease associations only work well if we have a good understanding of the background populations. So our failure of genetic association may be unique, dependent on unique structure of the populations. And rare polymorphisms, we haven't studied that in much detail. Maybe that's what we need to be doing it. We need to be thinking about epigenetic signatures also, because those may be unique and that needs to be picked up. Again, with the help of anthropological anthropologists, we should be able to pick up those what you call historical events as well as nutritional events or other things, so that we are able to say with confidence, yes, these populations do provide us a good indicative outcome in the personalized genomics or personalized epigenomic analysis. And remember, I said there is a what we call missing heritability. And missing heritability is a term which is used very loosely, where we are saying that Jiva studies are not providing us enough clues about where the genetic variation is. Only maximum successful outcomes are 10 to 15%, or maximum of 20% of variability is indicative of Jiva's outcomes. In terms of our missing heritability, because we have focused in on common polymorphisms in the past, we need to focus on rare polymorphisms so that we can study that missing heritability. Other problem with missing heritability is that we have in the past focused mostly on DNA analysis. We haven't focused on what we call epigenomics or epigenetics. And epigenetics is basically a relatively loose term again, and you can focus on range of activities in epigenomics or epigenetics. And epigenetics is basically a study of what we call environmental changes which happens, or which environmental changes which lead to changes in our gene expression, and those gene expressions then can be studied whether they are happening because of our food habits or our physical activity or other aspects. So the next part of this presentation, I will be focusing on how we can modulate our epigenomics to help in terms of resolution of what we call inflammation. So remember, epigenomic analysis, most people focus on DNA methylation, but then there are acetylation as well as histone modifications one could study. But DNA methylation is the most common method currently in that process when you are dependent on DNA which is extracted from, for example, blood samples or tissue samples. So this is again highlighting that we can have our genes will express or will be suppressed depending on the DNA methylation level or acetylation level or deacetylation levels in a particular situation. And our epigenome is modifiable. And that why I'm saying it is modifiable, because we can modify it by food, and we can modify it by exercise, we can modify it by reducing stress, or we can increase our stress levels and modify our epigenome also. but food is probably the most common, and exercise is the most common, which one could use to understand how epigenome can be modified. So what we're doing here in our Loughborough University labs is to look at if our epigenome can be modified by genotype as well as by our nutrition. And SNPs are linked to nutrients as well as to exposure, and that will lead to changes in the performance. So, for example, one genotype, a person may show improvement, carrying another genotype, person may show no effect, and carrying a different genotype will show impairment of that activity, either in health or performance. So I said that we can modify our epigenetic modifications, and we can do that by virtue of exercise, diet, alcohol, chemicals, microbiomes, hormones, cytokines. And basically, if you look at exercise and diet are the one which I'm going to focus today, and carrying out a range of studies which in our laboratory we do to see what effect we have in terms of changing our exercise regime or diet regime. So when I'm talking about DNA methylation, DNA methylation can happen at many levels. So it can happen at the promoter level, and it can happen at the gene body level. So in an unmethylated state, for example, there is these empty circles here. Unmethylated stage, all the CPGs are unmethylated, and either they are present in the promoter area or in the gene area. But in unmethylated stage, the protein will be produced normally, and it will be of what you call appropriate size and appropriate quality. If there is a promoter methylation, so promoter methylation is a major problem, and that means transcription start site will not function because of the methylation binding protein will bind, and it will stop the transcription to happen, and that means no protein will be produced. So this is what we call methylated state. And in gene body methylation, we have basically transcription site who functions, but its products are of different sizes or different amounts are produced. So that outcome is of concern in that process also. So DNA methylation, as I said, we will study on at two levels. Can exercise and food change the epigenetic signal? That we have been doing these studies for the last 5 years now. And so we can have different kind of exercises. We can have exercises which are carried out at aerobic level or resistance level. Resistance level means weights exercises. Aerobic will be running or bicycling or on the treadmill, etc. So we want to look at what effect each of these exercises has, whether they work independently or they work in conjunction with what we call food supplements. And the food supplement we are interested in are omega-3. Omega-3s are helpful in resolution of inflammation. And if you think about exercise can cause inflammation, so we are looking at whether exercise is inflammatory or exercise is a resolution of inflammation. Remember that a lot of people are using exercise as a medicine, and exercise as a medicine basically means there are benefits of exercise in helping us to achieve our physical as well as mental health. So we are interested in what effect it has on the methylation. So we are focusing here only on methylation, but one could study acetylation as well as other aspects, but depends on the choice of materials which you may have. So whether methylation goes up or methylation is removed, that's what we are trying to assess. So kind of studies we have carried out. For example, we have carried out a study on aerobic exercise and DNA methylation. And here is a outline of a 13-week trial. So we carry out these studies in the laboratory. And here we have a first week in terms of exercise trial, and then a supplementation period, and then no supplementation in between, and then supplementation with omega-3, and measure their what you call whether there is a benefit of omega-3 or olive oil in terms of exercise performance or DNA methylation. So DNA methylation, in these kind of studies, we take number of blood samples. So blood samples are taken before the exercise happens or before the study starts, and then at number of those levels so that we are able to assess them. So here is an example of what we call global methylation. So global methylation is basically all the methylation or average methylation for that particular cell type. So here we have taken blood samples and we have extracted DNA, and we have measured global methylation and also measurement of what we call gene expression of methylation transferases in that process. So DNMT1, DNMT3A, DNMT3B. DNMT1 is the maintenance gene, whereas DNMT3A and 3B are the they can cause methylation or they can cause demethylation in that process. So if you look at global methylation, pre-exercise level of percentage methylation is around about 79%, 79.2%, and after exercise it goes down, and whereas post-exercise in terms of DNMTs, again it goes down in that process. Whereas there is no major difference in, for example, DNMT1 gene expression or messenger RNA expression. Similarly, another gene, PPARGCA1A, and this one has, again, before exercise, it is much higher level. Post-exercise, it's much lower level. We expect that to happen. We are expecting that with exercise, methylation should go down. In most genes, that will be the benefit to the individual, because lower the methylation, higher is the chance of that gene expression or protein outcome. And in that process of post-exercise, methylation is considered as a outcome. IL-6, on the other hand, we don't see any major effect going on. Even with, for example, omega-3 or olive oil, we do not see a different effect or on the IL-6 gene. Remember, TNF-alpha, IL-6, and are the two main inflammatory genes one could study relatively easily in that process of understanding how how DNA methylation can affect the resolution of inflammation. So we carry out that what you call correlations also, and when we look at the correlations, we have a positive correlation between DNMT3A and TNF-alpha, whereas PPARG we have negative correlations and negative correlation with IL-6, between TNF alpha and IL-6, negative correlations. Each of these are significant. Apologies for interruption. When we look at effect of omega-3 on DNA methylation, we find that omega-3 does reduce methylation, but it's statistically not significant, but it is a significant decrease in that journal process. Whereas when we look at extra virgin olive oil, the differences are much smaller from the baseline. In terms of DNMT expressions, we expect them that N3-PUFA DNMT expression should go up, and that seems to happen there, as well as in what you call in our maintenance gene, which is DNMT1, which also goes up in that process. Similarly, we carried out what we call resistance exercise trials. And resistance exercise trials are, again, lab-based. And we take a different approach here. We took muscle biopsies, and muscle biopsies are taken from the leg tissue. And muscle biopsies allow us to test whether our DNA methylation is changed at the site of exercise. So whether that is happening at the site of exercise or not. And we also took a range of blood samples. Muscle samples or biopsies are limited to 3 muscle biopsies, whereas blood samples we were able to take every couple of hours and even after 48 hours of the exercise regime. So what we see a major difference going on between skeletal muscle and our blood samples. So what is happening here is pre-exercise TNF methylation is high. It goes down with exercise, but after 3 hours or so, it turns back to the normal levels. And it happens in trial A, trial B, trial C. Trial A is basically early part of that process. Trial B is after 3 weeks of omega-3 supplementation, and trial C is after 8 weeks of training, as well as omega-3 supplementation or olive oil supplementation. In leukocytes, we do not see a major patterning happen, and we have more data points here. Pre-exercise, post-exercise, 1 hour, 3 hour, and 48 hours. And when we look at the gene expression, we expect it to be the opposite trend, and that seems to be true in terms of post-exercise. TNF gene expression goes up, and that means DNA methylation is going down, gene expression is going up in that process of activity. IL-6, on the other hand, doesn't show much major effects going on, both in skeletal muscle and leukocytes. And in terms of focus for our further studies is going to be on different kind of ILs, because we find that IL-6 is much more a systemic cytokine. So we will be focusing on maybe alternative IL-6 molecules in that process to see whether ILs are influenced by DNA methylation or not. Correlations in terms of DNA methylation, we find that TNF is correlated negatively with messenger RNA as expected, whereas IL-6 is not showing major. Dark colors are basically showing no significant correlation. Blue ones are negative correlations, and red ones are positive correlations. When we compare with our exercise performance indicators, we find that TNF in skeletal muscle, remember skeletal muscle is the biopsy based, and that means they have been done at the site of exercise, and those exercises so those exercises are, and if you look at the leukocyte is much more in terms of blood based. So there are positive correlations with leukocytes, whereas at the skeletal muscle, these are negative correlations between our TNF and exercise performance or cytokines or muscle damage markers. PPARG, methylation and gene expression, and this is pre-supplementation, basically means before olive oil supplementation, which is in the dark color, and post supplementation is in the light color, and each of them basically showing that it does go down, but then it goes back in that normal process of uh, supplementation, that all omega-3 supplementations are helpful in that process. So in terms of methylation studies, we find that aerobic and resistance exercises do change methylation. And that means it can resolve inflammation. So both type of studies can help in a resolution of inflammation, and these should be recommended in cancer patients as well as diabetic patients, heart disease patients, that exercises should be carried out in a controlled environment or controlled process. The last study I'm going to focus today is basically looking at what we call what happens to telomere lengths. So telomere lengths are basically our aging biomarker. Shorter telomeres are associated with cardiovascular disease and diabetes and cancer, and inflammation and oxidative stress are the two major factors to accelerate telomere attrition. So we have carried out a 12-week low resistance, high repetition resistance training, and we have measured their what you call grip strength, counter movement jump, isometric leg strength, and we have carried out DNA analysis and RNA analysis, measured telomere lengths, gene expression, and measured inflammatory markers, CRP and IL-6. All these markers are helpful in understanding how inflammation or inflammatory markers can be modulated by exercise. So looking at our outcomes, we have reduced TNF alpha levels post 12-week exercise. So in other words, exercise does help to reduce inflammatory markers. And our telomere lengths in those individuals who are sedentary, sedentary basically means they do not have much activity going on, their telomeres go down in size from 0.819 to 0.734 at 12 months recall. And in terms of what is happening here is these individuals will get higher possibilities of disease or higher possibilities of heart disease and cardiovascular activities in that process. So in terms of to achieve better health outcomes, we need to kind of focus on genetics, we need to focus on environment, we need to focus on epigenetics, and we need to focus on exercise and physical activity along with telomere length so that our health outcomes are much better in that process. So holistic study needs to be carried out. It's not any good to understand simply genetic variation. We need to be carrying out a range of those activities so that we are able to say with confidence this disease has uh, helped in understanding of epigenomics or genetic variation in that process. So finally, in terms of this is again collaborative work with many colleagues from Loughborough University, Newcastle University, Dr. Singh, Dr. Puneet Singh from Punjabi University, Lucknow University, Cincinnati, Estonia, and thousands of participants in many studies which we have carried out, and many funding agencies which we have depended upon to carry out all these kind of activities in our research. Thank you very much for listening. I hope I have been able to give you some indicative understanding of what is happening in our field, as well as what are the potential possibilities. Again, as I said, we need to focus on a range of those activities collaboratively so that we have a better understanding of that process. We need to have kind of a holistic approach to our understanding of disease, holistic approach of to understanding population variation. Thank you.
Thank you, Dr. Masana, for your very extravagant and scholastic presentation. I'm getting huge response from our YouTube platform. Many students have asked very interesting questions. With your permission, I'm going to take one or two randomly selected questions.
Sure.
How much time do you estimate that personalized genome profile chip will be ready as today's Aadhaar card? This is a question posed by Navjot Kaur, our MSc student.
So say it again. I mean, what was your question?
How much time do you estimate that personalized genome profile chip will be ready as today's Aadhaar card?
Okay. Again, I mean, it will depend on the governments, and it will depend on the what you call willingness of the our system, whether we will have Aadhaar card based or Aadhaar card related genomic chip activity going on. But at personal level, somebody can get their sequence done immediately now. It will cost us around about $500 to $600, but that's possible. So it will probably be in the next 10, 15 years that we will have very clear understanding of that this is important, that everybody should know their genome and everybody should have the risks estimated. The main issue will be how we analyze it, whether we have enough bioinformatics tools, whether we have enough GPs or general practitioners understanding that genomic medicine is such a thing.
So, sir, the second question is by Keshav Joshi, our research scholar. He said, sir, what should be the right approach for drug repurposing or drug repositioning for personalized medicine?
Again, I mean, it depends on when that drug was identified and what targets or what receptors that drug is suitable for. And depending on that, whether we have enough genetic variation in those receptors or those areas of interest, and then we can identify whether there are other receptors or other binding proteins which may be of interest that could be used for repurposing. At the moment, I think there is a lot of talk about it, but I think the successes are relatively poor. That's my understanding.
Okay. So let me quickly take one or two questions. This question is by Sandeep Sharma. Can we predict differential expression of genes by analyzing DNA methylation?
Theoretically, yes. Practically, we do need to kind of consider a combination of protein level as well as uh, unless we have kind of tools which are very robust, we will have limitations.
Okay, sir. Another question is by Samoni. She wants to know, is there any computational method good in identifying DNA methylation regulatory sequences?
There are a number of computational methods available, but the main limit, again, as I said, I mean, if you're looking at genome-wide epigenomic analysis, then most of them are focused on already identified CPGs. So there may be many CPGs which may not have been identified in that process. So there are some developments there, but I don't think we can say with confidence that yes, we can use them as a starting point.
Okay, sir. Thank you very much, Dr. Masana, once again for
Thank you.
sparing and sharing your valuable moments with us. Now, while wrapping up today's proceeding, I request Dr. Ginjadhar Kaur to please propose a vote of thanks. Dr. Ginjadhar Kaur, are you there?
She's on mute.
Just a moment. Is it fine now?
Dr. Sahab,
It's unmuted now? Fine?
Hanji, your picture is not in this.
I don't know. Otherwise, the video and the mic both are on, Dr. Puneet.
Okay, go ahead, Dr. Sahab.
Okay. A very good afternoon to everyone. Respected Vice Chancellor, Sir, Professor B.S. Guman, Dr. K. Thangaraj, Dr. Sarabjit Mastana, Dr. Vasudha Sambhyal, our head of the department, Dr. Rajinder Kaur, Dr. Puneet Pal Singh, and all the other colleagues and the audiences who are here in this the Department of Human Genetics, place my heartfelt gratitude to our Vice Chancellor, Sir Professor B.S. Guman, who spared his valuable inspired us in many more ways to come. A deep sense of appreciation is with us through his video call, and in this moment, it was a delight to listen to him for his wonderful experiences for the standing up of this and department since last 50 years. My sincere thanks to the speakers of today's webinar, Dr. K. Thangaraj, Dr. research projects which they have done since many work was summarized before us and our students. And all this has made a plethora of knowledge being added to our knowledge today. And in depth, I would like to say that even most of the students who have joined through the YouTube channel, they would have gained a lot insight into this subject. Also like to thank the computer center people of our university who has been very supportive for the success of this program. And last but not the least, I would also play my special thanks to our head of the department, Dr. Rajinder Kaur, organizing secretary of this webinar, Dr. Puneet Pal Singh. They have worked really hard for this project to be completed, and the wonderful audience and all the colleagues who have joined it today. So in the end, thanks one of all. Thank you.
It was a wonderful session experience. Really enjoyed the proceedings. Professor Rajinder, you are very fortunate department that I have almost I've attended all the entire session.
Thank you,
Earlier,
sir.
I mean, it was a very thrilling, rewarding, and very an experience which others should follow. It's a role model. It's a practice to be emulated by other. If you have if you have recorded the proceedings, please do share with others. I think you have recorded the proceeding. Please do share with others, so that as and when there is a celebration like this, you have I mean, your speakers were excellent. I mean, second to none. They were competing among self. They are competing with themselves as well. That is the beauty of this celebration. Normally, Golden Jubilee celebration are cosmetic in nature, but your quantity-wise, quality-wise, content-wise, second to none. Heartiest congratulations, Professor Rajinder Kaur ji, Dr. Puneet Pal ji, and my dear colleague Gijinder Kaur ji. And yes, you have also, Gijinder, you have also joined the proceeding. Thank you. I express sincere gratitude to 3 high-profile speakers. It was an international day for us because Dr. Sarvjit Mastana had joined from England. I think he was there when I was at Punjab University. He did his PhD during that period because I was there from 81 to 2017. It's during this period that this center where you did your PhD was opened when I was on the campus. Therefore, I don't know whether we have met each other, but I have heard, I have visited your center many times where you did your PhD. And heartiest congratulations to my team, Dr. Jinder Kaur, Dr. Puneet Pal Singh, and all other colleagues. Excellent show. I'm really proud of you as a team. Thank you very much.
Thank you, sir. Thank you very much.
Thank you so much, sir. Thank you, Ji.
Thank you, sir. Thank you.
Thank you, sir. Thank you very much. Leave. How do I leave?
Thank you, Dr. Mastana.
Thank you, Dr.
Dr.
Rajinder.
Sarabjit Singh.
Okay, thank you. Bye-bye.
Thank you, sir. Thank you.
Bye.
How do I leave? Leave. I don't know.